A very good afternoon, dear delegates. Welcome back to the day 5 of this online FDP sponsored by ATAL AICTE on artificial intelligence. In the afternoon session, we have with us Dr. Prabha, who is a professor and head Department of Pharmacoinformatics, Naipur Mohali. I take a few moments to give an introduction to you. So Madam has done her BE and MTech from IIT Roorkee and PhD from Thapar University, Patiala. She works in the area of artificial intelligence, database development and data analytics. Ma'am has to her credit more than 65 research papers published in peer-reviewed journals and has developed approximately 20 softwares and databases. She's delivered 15 invited lectures, guided 5 PhD and 60 master students. We now have Dr. Prabha with us to talk about applications of artificial intelligence in drug discovery. Ma'am, over to you.
Thank you. So, as introduced, myself Prabhagar, I'm working in Naipur. So, today's presentation will be basically, I'll be dividing into the 4 parts. The first part where I will be talking about in brief when we are developing the AI-based models, what are the steps we are following. So I'll be just talking about in short about those details that what we follow as the instruction. It is not only the algorithm on which we have to work, it is the data preprocessing and the data compilation also. Then I will be talking about the one application on which we have worked or we have developed the artificial intelligence based model for the prediction of the blood-brain barrier permeability of the small molecules. The second application is for finding the protein function in the form of the subcellular localization, and third is the finding the interaction between the drug and the protein, whether there is some interaction or not. So I'll be talking about these 3 applications where we have applied this artificial intelligence in the Naipur. So to start with, when we are talking about the application of the artificial intelligence, let us talk about what is the intelligence we are talking about. So normally you compile the lots of data. So this data is comprised of the basic, unrefined and generally unfiltered information. All of you are doing the using the e-commerce or maybe the banking or any other applications. So the each one of this transaction is basically the raw data, which does not give you any useful or the interpretable information. But when you compile all the transactions or all the information related to some application, it gives you the information. So information is much more refined data that has evolved to the point of being useful for some form of analysis. So when you compile the multiple raw data to extract some information or some interpretation of that, that is known as the information. When you further apply this, or the knowledge resides in the user, which happens only when the human experience and insight is applied to the data and the information. That is known as the knowledge. And intelligence is the application and integration of the knowledge. So when we are talking about the intelligence, which is the application and the integration of the knowledge, if this intelligence is applied through the machines, that is known as the artificial intelligence. In your day-to-day routine also, you are using number of artificial intelligence machine. You may be using the smartwatch, which gives you number of information about you. You may be using a smartphone. You may be using the smart cameras. Number of devices at your home also are based on the artificial intelligence. So it is not only the drug discovery area, any area you think of today, you will find some kind of the application in the of this AI in that area. So while we are talking about this, for example, this is some data I am showing you about the gene expression data. So whenever you are having this microarray or the gene expression data, so how a specific gene is being expressed for a human being is you calculate by using this gene expression data. It does not give you any information if you look for a single or the individual sample. But when you collect the number of samples, you can find out whether in a normal to the disease condition, which genes are being upregulated, which genes are being downregulated. So that you compile this and then you find out the information, okay, these genes are being showing some expression in the disease condition, whether upregulated or the downregulated. Based on the knowledge of the user or the past experience, then they try to find out whether these genes are really making a difference for the specific disease condition or not. Then you apply to identify, like for the medical devices or maybe for the identification of the disease, you use this information. That is the intelligence you use. So using the machines, we try to apply this type of the intelligence in the normal or maybe in a specific scientific or maybe the engineering area. So, sorry. How we define the artificial intelligence is, this is the capability of a device to perform functions that are normally associated with the human intelligence. So if you train or you write the algorithms so that it can work as the human beings are working with their intelligence, then it is known as the AI machine. So AI is the study of how to make computers do things at which at the moment people are better. So AI is the science that automates intelligent behaviors. It is a system that thinks and acts like humans and rationally. All of you might have heard about the automatic cars also, where there is no driver. So some training or the intelligence has been built into these machines for driving. So these are the self-driven cars, are the part of this AI machines. So when we are talking about again this AI, it is the simulation of human intelligence by machines to solve the problems, to act rationally, or to act like humans. So it includes learning. When you are developing this type of the machines or the simulations, it includes the learning and the application of the rules for using this data or the information, as well as you apply the reasoning also within this AI-based models. So there are the different perspectives of the AI. There are the cognitive science, a way to understand the natural minds and the mental phenomena, or the mental action or the process of acquiring the knowledge and understanding through thought or experience and the senses. These are the areas where you can apply the AI. Philosophical questions you can answer using this AI application. Engineering, build intelligent machines. Science, you can use in any one of the field of the science, not only the drug discovery, whether it is the chemistry, physics, or the aeronautical science, or any other area you take, you can apply this artificial intelligence in that. Along with the artificial intelligence, we are also talking about the machine learning. So I'll be talking about few terms also. So basically machine learning and the artificial intelligence, you can say they are synonyms. Some of the times they can be used as a synonyms, but some of the time they are not exactly the synonyms of each other. So machine learning is an approach to achieve the artificial intelligence. You may or you may not, you train the machine using the ML algorithms to achieve the artificial intelligence that utilize computers and mathematical algorithms for building the models. Machine learning can also recognize a complex combination of the variables that reliably predict an outcome. Whenever you are developing any machine learning using any machine learning algorithm to build a model, basically you try to find out the predictors, the independent variables which you will use to build the model. Selection of these reliable variables is a major task while you are working with the artificial intelligence or the machine learning algorithms. Machine learning is useful for analyzing large heterogeneous data sets such as genomics or the epigenetic data. ML can be categorized into supervised, unsupervised, and the reinforcement learning. When we are talking about the supervised or the unsupervised learning, it is based on the training time, what type of the data you are using. In the supervised learning algorithms, you have the input as well as the output to train the system. So if this is the input, this should be the output. If you are using that type of the algorithm, then it is known as the supervised algorithm. The classification algorithm comes under this. or the function approximation algorithm like the ANN is the part of this, PNN is the part of this. If you are using the SVM or support vector machine algorithm, they are the part of the supervised learning algorithm. Unsupervised learning algorithms are when you do not have further training. If this is the input, you do not have the output variable for training also, then it is unsupervised. And the type of the problem which we use with this unsupervised learning is basically this clustering algorithms, in which you try to identify which are the similar type of the input data. And the basic concept of this clustering algorithm is that you try to find out if the two samples are similar, their action or the behavior must be similar. So you are broadly working with the supervised or the unsupervised learning algorithms. These are the steps which we follow when we are developing the AI-based algorithms or when we are working with the machine learning algorithm. First is the definition of the problem you work with. You have to define the domain of the problem or exactly what you are trying to find out. Unless until you are very specific what you want to predict. Just 1 minute. When you want to predict, that is the problem you have to define. Based on the problem, you have to identify the required data. If your data is not covering your problem domain, so whatever the algorithm you use, whatever the data pre-processing you do, you are not going to achieve a good model. So the selection of the data which is covering the problem domain is again a very important task. The data which may not be relevant and the variables or the descriptors which you are selecting may not be relevant for the problem, then again you will not be able to develop the model. Then the pre-processing of the data is required. I'll be showing you some of the steps for the pre-processing. Then the division of the data set into the training, test, or maybe the validation data set. After dividing this data set, the algorithm selection is required, whether you want to do the classification, clustering, or the function approximation, then you train the model. This trained model, every time you try to evaluate with the, you evaluate with the test data set. And if this test data set is performing well on the developed model, you can say the model has been built. Otherwise not. But not necessarily, again, I'm just explaining, these are the developed model which are giving a specific accuracy. Every data point may not be predicted 100% correctly. So these are the model which are coming with some kind of the accuracy. Some errors are always there. Nowadays, you may be hearing the term the data science, which is the extraction of the actionable knowledge directly from the data through a process of discovery, hypothesis, and the analytical hypothesis. And when we are working about the data science, it is basically a combination of the multiple disciplines. You need the programming skills, you should know the statistics, you should know the algorithms, the domain expertise for which you are trying to do the analysis of the data. So in the data science modeling, or whenever you are saying the machine learning algorithms, or maybe in the case of the artificial intelligence based application, the most common type of the application you work is the scoring, predicting or estimating a numeric value such as a price or a probability. For example, you are trying to predict the IC50 value of the molecules. You are trying to give the scores that this is the IC50, 1 nanomolar, 2 nanomolar, or micromolar, or whatever be these values. Or all of you are scoring the marks in your subject. That is the kind of the algorithm we use for the in this scoring. The ranking. You try to prioritize the molecules which may be more potent for a specific therapeutic category. That is the ranking you are working with. Learning to order items by the preferences. If you see in your class also, the students are being given the ranks. Class, first, second, third, and like this. So similarly, you also prioritize the sample data. Whatever be the application you are working, maybe you are trying to find out a potent molecule for a specific target. Maybe you are trying to find out the most expressed gene in the gene expression data, or maybe you are trying to find out the function of a protein for a specific therapy. So these are the ranking methods. Another is the clustering, which I have told you that you are trying to find out or trying to group the similar data, input data. That is the clustering algorithm. Here you can use the k-means, the hierarchical clustering. The number of algorithms are there which can be used. Classification, when you decide if something belongs to one category or another. Like in the classes or the departments, you are being classified. The student belongs to this class or this class. Similarly, the input data you can define into the various classes. Finding the relations, that is the function approximation. You define ax square plus bx plus c is equal to some value. That is the function approximation. By using the input variables, you are trying to get some value for the output variable. Characterization is the general plotting and report generation from the data. You are trying to find out how this data looks like. Plotting maybe the histogram, maybe some bar chart, or maybe some XY plots to just check what type of the behavior or what type of the data is there. So these are the few categories of the analysis we perform on the data, or we develop the model based on these criteria. In the data analytics also, basically we work with the 4 categories of the applications. The descriptive, what is happening? You do not try to analyze why it is happening, what is happening. Number of times you are hearing the polls on the TV. Maybe before the elections, you try to find out the poll results. That is what is happening. Any type of, they say, the inventory control you want to find out, or maybe at the time of the this COVID, you are trying to find out that the number of patients are coming to this. So you're just saying that what is happening. You say that there is an increase in the number of patients in the hospital. You do not try to analyze why it is happening. That is known as the descriptive. When you try to find out or diagnose this, why is it happening, that is a diagnostic type of the applications you work with. Predictive analytics is when you try to predict if this is happening, what can be the action you have to take, or what will be the output or the interpretation of the data. Prescriptive is what you should do so that you can achieve a specific goal. So these are the another way of doing the data analysis in the 4 categories which we define. Now, let us talk about the artificial intelligence application of the machine learning models which we develop. So the first step, as I have given you in the data, what are the examples of the data you are having? Whether it covers the whole problem domain space, whether it is not coming, or whether it is having some kind of a biasness in the data, everything you have to analyze by using this. What are the examples? What characteristics distinguish the class examples? Which features you have to use? There are the number of softwares. When you are having the molecules data for prediction of the IC50 value, you can convert them into the descriptors or the variables. Say molecular weight, maybe the polar surface area, maybe some C log P, or the other characteristics you can convert. So that is the example, the features or the variables or the descriptors which are relevant to a specific problem. Selection of those descriptor is again the important. Then you have to apply the algorithm, and then you have to do the validation of the data. So after you do the collection of the data, you have to do the data pre-processing. Before you apply the algorithm, the data pre-processing is required. So this data pre-processing is a data mining technique that involves transforming raw data into understandable format for further processing. Data preprocessing is used based on the maybe the data base driven applications, or in general all the data you have to pre-process before you apply the algorithm. In the data pre-processing also you have the number of steps. Data cleaning. When you collect the data, you will find out there is some of the noise in the data, some of the values are missing. For some of, when you will convert the molecular structure into the descriptor, the software, for some of the structure, they will say they cannot calculate 1 or 2 variables or the descriptors. So you may be having some missing values. There may be some conflict of the evidences when you collect from the multiple sources. One molecule may be defined as the inhibitor, and from the another data set you may find it is a non-inhibitor. So there may be some conflicting evidences, there may be some kind of the other noise in the data that you have to handle before you start building the model. The data integration is the another step. While you are collecting the data, again, I'm taking the example of this IC50. For some of the molecules, the IC50 may be, the value may be given in the IC50, in some of the, maybe the LD values or maybe some EC50. There may be the different formats on which that values have been given. So, but for building up the model, you have to bring them on the same platform. You cannot use the different representation to be used for the model building. You have to bring them into the same data representation. Then is the data transformation. The normalization of the data is required. In the normalization of the data, for example, when you are converting this molecule into the descriptor, the molecular weight may be having from 100 to say 6 or the 700 Dalton value, while the log P value may be say -342 say plus 2,3. So if you see the range of the values for these two descriptors is very, very different. And if you will try to build the model without doing the data transformation or the normalization, the model will be biased with the values with the higher range of the. So all the descriptors or the variables you have to bring within the same range. Maybe you will convert them into -1 to 1, maybe 0 to 1, or maybe say -100 to 100, whatever be the normalization you want to do, you have to convert them into the same scale. Then only the data, this classification or the function approximation algorithm works better. Then is the data reduction. When you will use any one of the software to convert this structure into the descriptor, they will give you the thousands of the descriptor. And your data set size is maybe the 100 molecules. By using that, you want to build the model. You cannot build the model with the thousands of the descriptors and only 100 molecules data set. So the sample size is 100 while the variables are thousands. So selection of the relevant descriptor is the part of the data reduction. The another data reduction is done based on the training and the test data set. You have to divide this whole data set into minimum two parts. One is the training, another is the test data set. So once on one set you are trying to build the model, from another set you will validate the model. So the data reduction is the again the part of the data preprocessing. Data discretization you have to perform. For example, you are working for the patient data, for building some of the model on the patient data. The age of the patient. You never use the age of the patient as exact numbers for as a descriptor. You always try to convert into some groups, maybe say 0 to 14, then 14 to 25 or 28, then the another group, then the another group. So depending upon the application and the type of the descriptor you are using, sometimes you have to convert them into the specific, this data discretization you have to do, or you have to reduce it to the few values. So when you have to do the data cleaning, for example, if you are having some of the missing values for some of the descriptors, one is you can ignore those rows or the data samples. Another is you can ignore that specific descriptors, but only out of say thousands of the samples you are having the missing value for 2 or 3 samples. Whether it is good to remove the rows or whether it is good to remove the columns. That depends upon the algorithm problem you are working. And sometimes you try with both the options and then you make the final decision with, which model is giving you the better output. The another way of, rather than you remove this rows or the columns, you try to fill these values by the mean value of the same class of the data, or maybe the median value. So again, in the artificial intelligence, the expertise of the person or the experience of the person makes lots of difference how the problem is being solved or being handled. It is not as simply that you insert the data, use some software and try to build the model. So any one of the decision may be taken. That depends upon the problem and the data size and the what type of the values are missing, whether it is important or not. Sometimes you have a pre-knowledge also that maybe the C-log P is very important for a specific application. So you cannot remove this descriptor for the model building. You may be removing the rows, or maybe you are trying to fill this value. In the data cleaning also, the outliers. Some of the descriptors are having a very, very significantly different value than the other observation. So normally they are being taken as the outliers. Outliers will make the biasness in the model, and you will not be able to get the good model with the outliers. But it is not always that these samples which are having a significantly different values, they are the outliers. They may be having a novelty in the data. They may be giving you some another information which may exist in the data. So again, it requires the analysis of the data, whether they are simply the outliers you have to remove before the model building, or whether it is showing some type of the novelty also in the data. So when in the data transformation or the normalization, there are the various methods which are available. The min-max scale, where you convert the value between the x minimum to the x maximum for each descriptor. There may be the decimal scaling, where you are converting between the 0 to 1, all the values, or maybe 0 to 100, whatever be the range, by simply dividing each descriptor value with the 10 raised to the power k. Or maybe you are using the z-score method, x minus mean divided by the standard deviation. So how your data is distributed, if it is having a kind of a normal distribution, you may be using the z-score. But if your data is not having a normal distribution, maybe you are using the decimal scaling or the min-max scale method for the normalization of the data. So in the data reduction, one is that you are trying to divide the data into the training and the test set, and the another application is that you are trying to remove the irrelevant descriptors. So for the feature or the variable selections, there are the various algorithms. And when you are using the algorithms for the feature selection, never use a single algorithm. You use multiple algorithms and see what each algorithm is giving you the important feature. None of the algorithm you can say it is the best or this is the final result which gives. So you have to use the multiple algorithms and see what is the common attributes are being given. And sometimes you have to use the hit and trial method by using the permutation combination of the list of the descriptors given by this algorithm. So the algorithms which are being used, like using the filter methods, maybe you are using the percent correlation method where you're trying to find out the correlation, or maybe the LDA or the linear discriminant method, ANOVA or the chi-square. I'm not going into the detail of these algorithms. You may be studying in your statistics or maybe somewhere else. But these are the methods which you can use as a part of the filter method. Or in the feature selection, there is another category which is used as the wrapper method. Now in the wrapper method, you train the model with each iteration before adding or removing a descriptor into the final selection. Again, in this, you have the forward feature selection, backward feature elimination, or the recursive feature elimination, or maybe the Borota algorithm. So there are the number of algorithms which you use for the feature selection or for the final elimination of the irrelevant descriptors. So whether you use the forward selection or there's a backward elimination algorithm, again I'm repeating that you should not use a single algorithm for the feature selection, because if you have not selected the correct set of the features, your model will never give you the good accuracy. That is the big. So these are some of the algorithms which you can use. I'm not going into the details of this. Then the last step is when you have built the model, you have to do the validation. For doing the validation, also there are the number of ways you have to do the validation. Not simply training set and the test set. When you are dividing the data set, On one set, you are building the model. Maybe sometimes you divide into the 70-30 ratio or maybe the 80-20 ratio, again, depending upon the size of the data set. So this test set you try to use for the validation of the model. Because whenever you are trying to use any one of the algorithm for this AI or the machine learning algorithm, few parameters you have to optimize. For example, if you are using a KNN algorithm, K nearest neighbor, in the K nearest neighbor, the value of the K, how many neighbors? Maybe you people are not aware of this KNN algorithm. So this is the algorithm which we use for the classification, and we try to find out the new sample data is near to which already known sample. So K is the value which we define how many nearest neighbor we are going to consider. And for finding out the nearest neighbor, we use the distance metric also, whether it is a Euclidean or a Manhattan or a cosine, which metric you are using for the distance also. So with each algorithm, few parameters you have to optimize in machine learning algorithm or in the artificial intelligence algorithm. For example, if you are using the artificial neural network, there the number of layers you use, the number of hidden layers, and the number of neurons in each layer you have to optimize. Every algorithm, one or the two or few variables you have to optimize. So with all these variations, you have to check with the test data set and the other method, which gives you the optimum value for these variables. So for doing the performance estimation or for the data validation, you try to use this data validation method. Now in the data validation method, one I have given you that you use the training and the test set. While you are optimizing this parameter, you try to build the model. If you do not use this test data set, there will be always overfitting on this data set. So you try to find out at each iteration what is the performance of the test data set. Based on that, you try to find out the stopping point for the optimization of these variables. The other method which we used is the k-fold cross-validation or the leave one out cross-validation. In the k-fold validation, what is being done? That whole data is divided into the k-folds or in the k parts. K is normally taken as the 5-fold or the 10-fold cross-validation. Now in the cross-validation, if, for example, if we are talking about the 5-fold cross-validation, data is divided into the 5 subgroups. What is done in this, 4 subgroups are used for training of the model and the 5th group is used for the test set. Then in the next iteration, you will include this test set or the one sub data set into the training and the another one you will remove. So in the 5 iteration, every time one subset will be used as the test set and the remaining 4 are used for the development of the model. And the average accuracy from all these 5 models which are built, that is being defined as the 5-fold cross-validation. So again, you are not working with only the training and test data set. You are also doing the 5 or the 10-fold cross-validation also on your data set before you say your model is good to predict the value. Again, you will also work with the leave one out cross-validation. Why the leave one out cross-validation is performed? Sometimes maybe some outlier is there or maybe some kind of a biasness with a one sample which may be affecting the performance of the whole model. Maybe it is just improving the parameter, say the R square, or it may be reducing this value to a very low value. So you just use the leave one out method and see whether the this cross-validation is giving you the good results. Another very method is used as the wide randomization method also. Wide randomization method means with the input variable, you will just jumble the values or you will just rearrange or randomly give the output value and then you try to build the model. And again, if your model is giving the good result, means it is a by chance model has been developed. So you do not believe on the model or the features or the data set which you have used for the model building. So the wide randomization, leave one out, or the 5-fold or the 10-fold cross validation, and this training test and the external test validation is required. And if your model is giving the good performance parameters based on all these validation methods, then you say your model is built and it can be used. Once you have said that the model is built and you want to predict with the new data set, again, there is an applicability domain. The new data set must be similar to the data on which you have built the model. That is your problem domain on which you have built the model. Some extrapolation is, it should perform, but you cannot have totally out of domain data, and you cannot rely the model prediction from this for this data set, which is not similar to the training data set. So there is always the applicability domain which is checked. For example, there is a gene expression data. For one gene, the expression values are written here. For second gene, the expression values are on this. The blue dots are showing the internal or the training set data, and this is the new data set in the red or the external data set. It is similar to this. If you see, it is lying in the same space where your training data set. So it is within the applicability domain. The prediction for this data set must be reliable if you have built the good model. So the applicability domain is another parameter of even you have built a good model, every data point you cannot predict from the built model. So now I'll be talking about the applications where we have used this artificial intelligence. The first application is for the blood-brain barrier permeability, where we have used the artificial intelligence, and basically the artificial neural network we have used. In the artificial intelligence also, it is not a single algorithm. There are the hundreds of algorithms which you can use for developing the AI-based model. So we collected the 191 molecules from the various data sources, and we have used the 7 descriptors. These 7 descriptors is based on the already known knowledge that from the literature we have identified that these are the descriptors which are available. So basically, in practical, initially actually we started with the 18 descriptors, but the 18 descriptors we have not we have not received the good model. Then we applied the data selection and finally converted into these 7 descriptors. These 7 descriptors are known in the literature which are affecting the blood-brain barrier permeability, like this molecular weight, topological surface area, number of hydrogen bond acceptors, donors, etc. In this first, we have checked for the 0 variance. Why we check for the 0 variance before we build the model? If any one of the descriptor is having the 0 or a constant value for all the data sample, means it is not affecting the output value. So having that specific descriptor does not make any difference or does not make any sense for using this descriptor. So the correlation coefficient, the highly correlated descriptors are also not used. You use the one descriptor. There are the 2 descriptors which are highly correlated. You remove one, which is having a lesser correlation with the output parameter. Then the splitting was done for this data set. 141 we had used in the training set, and the 50 we have used in the test set. And the decimal normalization we have used for the normalization part. So log BB value of all the molecules, because we have selected only those molecules which were having the log BB value. This is the output variable. The supervised function approximation algorithm, that is the artificial neural network algorithm we have used. In the final model, we were having the architecture 7521. The 7 number of neurons in the input layer, because we are having the 7 descriptors in the final. These are the 7. So whatever be the number of descriptors that is in the input layer. Then this is the part 5 and 2 is by some training using. This is the part which we have to optimize for the given data. And the number of neurons should not be total number of connection in this, means 7 into 5 plus 5 into 2 plus 2 into 1 should not be normally more than, ideally it should be 1/3, but in this case it was 1/2 approximately, so that your model building is good or the reliable. With each in the artificial neural network, the another parameter we have to define is the transfer function. Again, as I'm not talking about the architecture of the ANN, there is a transfer function we use in the artificial neural network. Multiple transfer functions can be used, like the log sigmoid, 10 sigmoid, linear function, or the other functions. In this case, we had used the log sigmoid and the linear function. Log sigmoid from input to this, and this linear from here to here we have used in the layer. And then we have used the MATLAB software for training of this model. After training, we found there are the 9 molecules which are not being predicted. The 9 molecules are showing a very, very different, and the final value of the model was very, very poor. So we removed this 9 molecules and used the 132 molecules for the bedding, and the model was good, having the correlation coefficient 0.9, and on the test set is this coefficient of determination like this. These are the various parameters which we have used for the evaluation of the model. Now on the 9 molecules, the outlier analysis we did, and we find out all of them were belonging to the neuromuscular blockers, except the 1 molecule, the Brazil. So they are from the 1, and if we were seeing their output value, the log BB value, that was very, very low or totally out of the range with the remaining molecules, where they were having a proximity up to -5 or 6 to the +5 or 6. They were having a very different values. Then we checked their input variables values. We have to analyze why they are showing this type of the very low log BB value. We checked either some of the molecules are having the slightly higher molecular weight, or their C log P values are low. So they are not able to cross the blood-brain barrier. For this molecule, Narcuron, we find out there is a PGP. It is a efflux transporter. This value for these 4 molecules is very high. So even if their rest of the properties are comparatively good, which is for a permeable molecule you need, based on the literature, these 4 molecules, when we have analyzed and identified, their PGP value is very high. So from the passive transmission, like they are being effluxed by the PGP. So the final log BB value, how much quantity of these molecules can cross the blood-brain barrier is very, very low. So even if you are finding that some of the molecules are the outliers, you have to analyze why they are outliers. It is not simply you just remove and try to get the very good R square and the other validation parameters and just do the analysis. So after doing the analysis, we remove because they are not the category of the molecules, and simply by just seeing these parameters, we can identify it and develop. So after this, the final model, which was developed with the 132 molecules, we finalized that. Then we compared the result with the other published work also, or the one of the software which is commercially available. And we found that this model is predicting whatever be the data set which we were having is better than the other softwares. In this work, we have further worked on this, though I'm not having the data for that. We found that the efflux is making, or these efflux transporters are making a difference in the log BB value. Then we use the BCRP, the another efflux parameter for the prediction. It has improved the value, and the MRP one also. That is another efflux transporter. So we use those 2 descriptors also for the prediction of the log PV value. So it is not only the development of the model. If you have the knowledge, like what type of the parameters or the descriptors which are making or affecting the values, you should consider that. So this is how we worked on the log PV variable, and finally we published this paper in the JCI. The another work which we have done on the subcellular localization prediction. In this subcellular localization prediction is we have used the primary sequences of the proteins. And when we have collected the data, we have used the UniProt database. And we have used the organism of the eukaryotes and collected the data, and we just removed the protein sequences or the hypothetical proteins which were having the sequences less, the sequence length less than 30 amino acids, or maybe having some ambiguous amino acids like B, X, and Z, which we have removed. And in this, we finally got the data set of 38,354 proteins. And on this data set, we applied a program CDH. So if the protein sequences are very, very similar, it will have a biasness in the prediction. Your data should be equally distributed in the problem domain. So we use this CDHIT program to remove the like very similar sequences from the data set. And the test sets data set, which we have used from a previous study from this researcher, which we used for the validation of the model we have built. Now to convert these protein sequences, which are all of you may be knowing, these protein sequences, a combination of these 20 amino acids. So we converted them into the 29-dimensional vector, the 29 descriptors. The first 20 descriptors is for each descriptor, we have calculated their frequency. What is it in the composition? What is their frequency for the each amino acid? If there are the 100 amino acid A is what is the percentage of that amino acid, and convert it into 0 to 1 ratio. So these are the 20 descriptors we have used, and the remaining 9 descriptor is based on their physicochemical properties like this mass, hydrophobicity, and the hydrophilicity. This amino acid index for these 3 properties, mass, hydrophobicity, hydrophilicity, we have collected from the net. Then we use the sequence order also. If after A, which amino acid is coming, and based on this equation, we have calculated the remaining 9 descriptors. So we got our data set, which is having the 29 descriptors. Around 38,000 sequences were there for the training of the data set. In this problem, we have used the 2 algorithms, K nearest neighbor and the probabilistic neural network. And in the K nearest neighbor, we have used the value of K, only the nearest sample is used for the prediction, and the cosine has been used as the distance matrix. In the second algorithm, we have used the probabilistic neural network and the Euclidean distance as selected. The purpose was using these 2 different algorithms was, if you look for the parameter here, we have tried to find out as the difference between the 2 vectors based on their angle, not based on their length of these vectors. If you say the 29-dimensional vector or the 29 descriptors, you cannot plot the vector in the 29-dimensional, but you can assume like you are working with the 3 dimensions. Similarly, if you have a space in the 29-dimensional space, how the vectors are given. So in the first algorithm, we are making use of the angle between these 2 vectors. In the second algorithm, we are making use of the distance between these 2 vectors. All of you have studied the vectors in your, I think, 10th or 8th class, where you know about the angles and the how to find out the distance between the 2 vectors. So these are the 2 different algorithms. In the K is equal to 1 means only the local effect, and in the probabilistic neural network, all the sample data is used for identifying this distance. So two different algorithms for the classification with the two different approaches were used. These are the parameters because it is a classification algorithm. So R square cannot be used for this. So the precision, recall, or the sensitivity, specificity, accuracy, or this MCC. This is the formula for doing the statistical analysis or the validation of the model were used for the model. If you see in this data, out of this 38,000, when we have analyzed for this subcellular localization, these many samples. But if you see for the lysosome, only 150 samples were there. Is there some kind of a prediction we were able to get with these smaller values also, like this accuracy? Accuracies were not that bad for the very smaller data set also in this, using the 5-fold cross validation in the KNN as well as with the test data set also. The accuracy was not very bad for the smaller data set. Similarly, with the PNN also, we tried to find out all the 5 statistical parameters and could find out even if the data is small, we are able to predict these proteins, the subcellular localization of these proteins. Then we did the further analysis on this data. As in the KNN algorithm, we have used the cos. And in the cos, basically, as a distance matrix, it uses the angle in the angles. So what we converted into the degrees and checked, done the analysis. And if we find if the new sample with the training data set is having the angle between 0 to 6 degrees, the accuracy is somewhere 85 to 90 between the 5-fold and the test set data set. Similarly, if the angle between the new data point and the training data set is between 6 to 8 degrees, this is the reduction in the accuracy. This is how the accuracy has been changed. So the how close the angle, so they are able to predict based on the angular. And this is what we have used this information or the knowledge to give the probability of the correct. Because all the subcellular localization were not in our training data set. So when a new protein sequence has to be given, it may happen that it is not from any one of these subcellular localization which we have used. It may be for some another subcellular localization. Then we are finding it is giving the more angle between the new protein sequence to the training data set, which is closest to this. So which was used to give the probability of the correct predictions in the paper. And this again we published in the in silico biology. This paper was published. So how we have given, if KNN and the PNN are both predicting the same subcellular localization, this is the probability has been assigned. If they are giving this and the angle is between this is the probability. This is the way then we have defined. So there is no method by which you can define that this is how you have to apply. The analysis of the data is required while you are working with the artificial intelligence, and the prediction and the inferences you have to identify. Initially, this model was developed on the single subcellular localization. Then we extended this on the multiple subcellular localization, the same thing. And we were able to predict the multiple subcell also with the probability. If for any one of the protein, if you see the UniProt data set, I don't know from the pharmacy how many of you have checked the UniProt database, but if you go there, a single protein may be having more than one subcellular localization also. And we were able to predict this also very correctly. The same algorithm we had extended on the enzyme class prediction also. And successfully we could apply that also. The third application where we have used the artificial intelligence also is the drug target interaction. In the drug target interactions, now we have to use the descriptors from the small molecules as well as the descriptors from the macromolecules, means the enzyme or the protein. So the descriptors from both molecules have been used and known as the proteochemometric algorithm approach in this. So while we are working with this drug target interaction, the data set was available that this small molecule is interacting with this given protein, but the data set was not available for the non-interacting pairs. Nowhere it has been defined this is the molecule, this is not interacting with this. No one test for that. Nowhere the data was available. But while we are working for the classification whether this drug target interaction is existing or not existing, we need the negative data set also. If you have only the positive, then how to identify when the drug and the targets are not interacting? So this was the challenge, to work with the non-interacting data set. Anything which has not been reported cannot be considered as the non-interacting. Someone may not, like no one has, may have not been tested, but it may happen that they are having the interactions between the drug and the target, some of this. So blindly we cannot use. So we devise one a strategy to identify or to create the decoy set for the non-interacting pairs. So while we are working, so there were so many databases which were available for the bioactivity data set. Initially, we started with the PDB data set. It was having the 3D structure also. So while we were working on this, we used a 2-dimensional as well as a 3-dimensional, like this primary sequence as well as the 3D structure was used for the descriptors of the proteins also. So we use this PDB data set for the collection of this. Now, when we have retrieved the data, we found out there are the 9,800. This is the number of interacting complexes existing in the database. The distinct proteins from this data set were 3,000 something, and the different ligands of the small molecules were 5,000 something. So ligands were inspected and prepared in the Discovery Studio. This is a commercially available software. We have prepared them. Prepare means their structures, their energy minimization, checking their, calculating the descriptors. For so many, they were built using the Discovery Studio. For the proteins, using this binding site, we have retrieved the complete sequence from the UniProt database. So the pairwise distance matrix was prepared for these PDB entries by using the Euclidean distance and calculated on the first K binding site fingerprints. Then the affinity propagation methods for the scikit-learn was used to cluster. Then what we did, we are having this many complexes. We used the clustering algorithm. And in any one of the cluster, if we are having less than 10 entries, we remove those entries. Used only those clusters for the predictions which are having more than 10 entries. We'll be finishing in 10 minutes. So the final data set for this were having the 3063 complexes with these many different targets and the 2040 different ligands. Then did the analysis, how these proteins were distributed among the various species. So we did the analysis, how they are having the enzyme class or the function, this EC numbers, their distribution among the various classes of the functions have been done. The resolution on the crystals were also done the analysis on this. Then we have also used the external data set. For developing the external data set, we have used the PDB data set for the training data set, but we have used this data set from the Campbell Drug Bank and this therapeutic target database for the external data set. External data set means you have not used for the training, and in any one of the step of the training, you have used this data set. We have, while we are retrieving the data set, this is the range of the small molecules, molecular weight has been taken care, and we have also used the 10-meter similarity coefficient with the training ligands also. Extrapolation is done, but not to beyond a limit. So 0.8 nanometer similarity has been maintained while we are retrieving the small molecules with this. And only those targets were used or will be used in this which we were having in the training data set. Now for generating the negative data set, randomly has not been selected. So what has been done in the training data set? For the training data set, it is for the chemical nanometer similarity has been checked less than 0. They have to be very dissimilar from the positive data set. Then only they may not be interacting, but their physicochemical properties must be similar to the training data set. Physicochemical properties means their molecular weight, their log p values, and the number of hydrogen bonds. So their physicochemical properties were similar to the training data set. So this is the criteria which we have used for the selection. Now, even after selecting, we have combined them the random with the proteins in the training data set. Then we have used the weighted SVM model. SVM is a machine learning algorithm for the classification which was used. The positive instances which we have collected from the database were given the more weight, and this randomly or created the decoy set were given the less weight, and built the SVM model. If any one of this negative instance which we have created were being predicted as positive, that were removed from the negative data set. And we have repeated this number of times, and finally we have generated the negative set. And using both this now, the positive and the negative data set, the descriptors, sequence-based descriptors, these many from this Prophet web server, the structure-based descriptor using this software were this many, and the Morgan fingerprints for the ligands using the AICTE were these many. This is the total list of the descriptors which was used for the model building. Use the SVM, RF, and KNN algorithm. Again, for the SVM, the C and gamma were optimized. For the random forest, the number of M-trie or the number and the number of decision trees were optimized. For the KNN, the value of K and the nearest neighbor, the distance matrix function was optimized. And finally, we could use the model with the different permutation combinations. These were the accuracy, and we found out there is no idea of using the 3D structure descriptors over the primary protein sequences, because it is much more easy to retrieve those protein sequences and converting into the descriptors. Then further we did the analysis on the natural products which are available for the cancer. So we predicted the targets for this Nedrolone for this drug, and find out these are the proteins with this score, the probability score, and this is the docking score. We identified the targets for this. All predicted targets for this belongs to the Homo sapiens and have been reported to play an important role in the cancer pathogens. So this is a natural product for the cancer, and we find out the targets for this and which we identify. So it is giving a more reliability of the model developed. Then not only for this, then we have used this protein, and we have identified that these are the drugs which are binding with this probability, and this is the docking score by which. So these are the predictions which were done. Similarly, for these two drugs also, the proteins were identified from which organism. So this has given, for example, in this drug for the sorafenib, these 7 targets were already known, but these are the 3 new targets we predicted that they are interacting with this drug. So this is how we have used the artificial intelligence for building the model. And then we published this work in the molecular biosystems. So it is not a single area or a very well defined, whatever the applications where you can formulate or you can use the artificial intelligence for the prediction, classification, or maybe for the clustering of the molecules, or maybe the proteins, or maybe the pharmaceuticals, whatever be the applications you try to use. So with this, I'm just finishing my lecture. I think I'm very well within time. There are 4 more minutes to go. So, but with this, I'm just finishing this lecture. So these are the 3 only on the small molecules, the interacting with the drug and the target, and only by using the protein sequence to predict their protein functions, we have applied the artificial intelligence algorithm in the machine learning, which we can use. So you can apply as a pharmacy student for any one of the pharmaceutical products for the classification and maybe predicting the PKPD or maybe PBPK properties. This pharmacokinetics and the pharmacodynamics parameters can be also be predicted using the artificial intelligence. So with this, thank you everyone. So now it is open for the questions. Any question?
Through. Am I audible?
Yes.
Yes, thank you, ma'am, for your presentation. We have a few questions in the chat box. Dear delegates, if you have any queries, please post your questions, or you may also use the raise hand option. So, ma'am, my query here is, you were talking about data sets. So, usually, what is the the size of the sets that we are talking about in terms of GB or TB? Because accordingly, we require the infrastructural setup to support this activity, right, of AI.
Data set depends upon the size of the data set depends upon the problem you are working with. For example, in the case of the blood-brain barrier permeability, we were having only the 131 molecules for testing of the data set. But the molecules, the similar type of the molecules we will be able to predict, not very, very different. But in the case of the drug target interactions, we are having in the lakhs of the molecules. Training set was in the around 38,000 or something, but the
yes.
External set which we have used, which was in the lakhs of the molecules, because the number of proteins which are available and which covers the problem domain is very large. So that depends upon the size of, that is the way we define the size of the data.
Yeah, based on the project objective that we are talking
Yes,
about.
and the availability of the data. But only thing we have to say is there should not be biasness. Very, very similar type of the data if we are using, which is not having a variation in the input data, it will create a biased application. I think there is one more question. Is it mandatory to test using negative data set? The problem which I have shown you is for the classification, true or the false. When we say between the small molecule and the large molecule or the macromolecule, whether the interaction exists or not. So when we are to training, we have to have a negative data set. Like in the subcellular localization, we are trying to classify into the, say, 16 subcellular localization. There is no negative data set. Based on the angle, then we try to find out if we are having a new protein which is not having any one of the subcellular localization, how we are going to identify. Otherwise, it will predict one of these subcell. So we tried to, we did the analysis based on the angle, that if the angle is more than this, between this, the probability is less than a given limit. So it is not from any one of this subset.
Okay, ma'am, now since this point has come, I think we had this Nobel Prize being awarded for this discovery of piezo receptor, right, ma'am? So if somebody has to start working on this, there is no data availability. I mean, as of now, I'm not sure, I can't comment on that. But if we have to begin it, so do you say that we have very less information to take tasks regarding that?
Somewhere we have to start. So, but initially, you cannot start without having some data. Initially, you have to have some knowledge. Maybe if you are having that, always gives you a better platform to start with. And the quality of the data, slowly it improves. Once any one of the area starts, initially you may be having the 100 samples, maybe the people start working, then you may be having the 150. So it is a kind of a cycle. You build the model, what is available, then you keep on improving the model based on the availability of the data.
Yeah, I think so. Wherever the data banks are being updated, from there we have to collect and start proceeding with the work.
So yes, it is always the cycle. You keep on updating, or you can improve the model.
Interesting, ma'am. So we have one more question here. If design molecules comes with an applicability domain, then can we proceed for synthesis of that?
Yes, if you believe on the model developed, you have validated thoroughly. We tried. Like in one of the applications, we have used the approved drugs for the repurposing of the drugs for the Alzheimer. And we have done the testing also. We got the good results also with the repurposed drugs. So, I mean, you have to have the faith in the developed model. And whether it is validated properly, because you are saying that the design molecule is within the applicability domain, yes. Ideally, it should, but these are the prediction which can never be 100% correct. Okay. If you take the 10 molecules, there are the chances, even if it is a very, very good model, 1 or 2 molecules will fail. Okay. I'm giving with all the hypothesis, if your model is very, very good, still there are the chances 1 or 2 molecules will fail. So don't get disheartened by this. Always try to improve.
So if I'm asked from your experience, were there any situations where the models did not perform as expected?
Actually, when we are working like with this initially, or the students are, that is their training phase. All the things have not been given with the all the consideration or with the maybe. But if you work with the artificial intelligence model and the applications you see in the real life, yes, not on a very small thing, it is on a very, very where the risks are very huge. The artificial intelligence based applications are successfully predicting, giving you the results. If you see a simple weather forecasting, the type of the prediction it was giving 20 years back, and the quality of the predictions it is giving nowadays has improved. Such a complicated system. The people may say in a very simple word, oh, it doesn't predict. But you see the complicacy of the whole system. So it is improving day by day.
Yes, yes. Thank you, ma'am. So any more questions from the delegates? So in the absence of any further questions, ma'am, we thank you once again for being with us this afternoon and sharing your insights and knowledge in this ATAL FDP on artificial intelligence. Thank you, ma'am, for being with us.
Welcome, delegates. Very good afternoon to one and all. We have the afternoon session with Mr. V. Ashwatharamaya. To say a brief resume about him, he is the head of Unique Consultants, Bangalore. He has 20 years of experience. He is an executive at MS Vidya India Limited, Bangalore. In 1992, he has set up a management consultancy known as Unique Consultants. He has conducted training for more than 4 lakh employees, frequently conducts training for delegates, professionals in Dubai, Hong Kong, Muscat, Qatar, Sharjah, and Kuwait. He has subjects of training in related to industry, HR interventions, communication skills, and presentation skills. He has conducted training and management with management associations like CII, FKCCI, NIPM, Red City, Bangalore, and Management Association. He has written books and CDs and authored books like a celebration called Life, Redefining Husband-Wife Relationship, Success Through English Alphabets, Great Personalities, etc. He has released audio and CDs on stress management, Spice Your Life with Your Spouse, Husband and Wife Relationship, and Formula for Success for Students. And he has produced a video film titled Parivartane. I request all the delegates to go through his presentation and have a wonderful time. And, this session is a mandatory session by AICTE. Hope it will rejuvenate yourself in your workplace. Thank you very much, sir. Over to you.
Thank you. Good afternoon to all of you. It gives me great pleasure to make a presentation on the topic of life skills, that is stress management and yoga. Life skills are a set of skills which are predominantly human oriented. Generally, to be very successful, we need two types of skills. One is the technical or the domain specific skills.
Excuse me, sir.
Yeah.
Just sorry to interrupt.
Yeah.
We need Naveen sir to upload the slide again. It's not in the loop.
Who, Mr. Navi?
Yes. He has to share a slide.
I'll do it, sir. I'll do it now. I'll do it.
I'm not visible here.
Yeah.
Fine, sir.
Fine, sir.
You can go ahead.
You can go ahead. Okay. 1 second. Now you can take out the slide, no? No?
You can proceed, sir.
Yeah. 1 second. My video is not coming here. That's why I'm just wondering.
We are
We are able to see you, sir. But require slides for your presentation?
No, I am not able to see. There is some.
We can see you, sir, but we are not able to see your slides.
I am not able to see. Let me see. 1 second. Yeah, no, it's okay. Shall I continue?
Do you require the slides, sir, for the presentation?
No, not now.
Okay, because we are not able to see the
When
slide.
it's required, I'll put it, madam.
Okay, sir. Right. Thank you. Fine.
Sure. So basically the topic is on stress management and yoga. Initially I'll be talking on stress management and then on yoga. Now, generally the stress has been defined in about 3-4 ways. One is whenever a person's body and mind exceeds the threshold level, we are under stress. Anything has got a threshold level. All of us do understand that. There is a limit to which one can withstand the pressure or tension or load. So if we are making our body and our mind to undergo more than the threshold level, the pressure or tension, we are said to be under stress. Clinically, stress can also be explained as the stimulation of sympathetic branch of nervous system at the cost of parasympathetic nervous system. In fact, our central nervous system gets branched out into two. Sympathetic nervous system, which is called as a fight or flight system, and there is a relaxing system called as parasympathetic nervous system. So if we continuously stimulate the sympathetic nervous system, but we don't relax it commensurately or equitably from the parasympathetic nervous system, we are said to be under stress. Now, against the common belief that stress means always bad. See, the moment we say there is a lot of stress in this work, it is normally used in a very negative connotation, that there is something wrong, there is some anxiety, there is some uncertainty, there is some unpleasantness or irritation, etc. But contrary to this belief, certain amount of stress is very much needed for our life. In fact, if there was no stress, the life wouldn't have been so beautiful or enjoyable. Certain degree of stress is very much needed, which of course is called as eustress, a good stress which is needed. But unfortunately, as we increase the stress levels beyond a particular limit, which is very, very subjective or characteristic of the human being, then the sense of well-being or the health or the performance or the optimism starts deteriorating. So which means if we can put a graph, as the stress increases, our performance, health and sense of well-being increases to a certain extent till it reaches a point after which it starts deteriorating. So we should ensure that we stress ourselves to that extent which is needed, but afterwards we should be very cautious that we don't stress our body or mind. So that stress is called as distress. It is something I always give an example of a violin. Suppose we take a violin and the strings of the violin is very loose, and you want to play violin, you cannot get good music. because the strings are very loose. Similarly, if the stress levels are very less in our life, life is not beautiful and life is not enjoyable. But at the same time, in a violin, if these strings are too tight, almost to the brim, and you want to play the violin in that violin, you want to play music in that violin, the strings will break. So beyond a limit, if we stress, there may be burnt out or breakdown. Then when will we get the most melodious music from the violin? When it is optimally stressed, neither more nor less. Similarly, we need to get ourselves stressed to an optimum level to get the best of our capabilities, our competencies, our sense of well-being, our performance, and our health. Stress can also be defined another way. It can be explained as the difference between the expectation and the reality. Imagine the expectation is here and the reality is here lower. The difference can be told as stress, which only means if we get in our life whatever we expect, theoretically speaking, there should be no stress. Then the lines will be like this instead of being like this. So it's very obvious if we have to reduce our stress levels, we can do it in two ways. One, either we can increase the reality, which means we can become more competent, enhance our professional skills, our capabilities, our endurance, whatever. Or the other way is, if we cannot increase our capabilities by any chance, the other way of reducing the quantum of stress is to reduce our expectations, which may not always be welcome, of course, because that may make us inefficient, that may make us callous, nonchalant, and take it easy. So, one way of managing the stress is either to increase our capability to manage the stress or lower or reduce our expectations. Now, the stress can be physical and also mental. In fact, our mind and body are the manifestations of the same entity. When the mind gets affected, it will precipitate into many physical disorders, which is called as psychosomatic. Psycho means mind, soma is body. So mind influences the body. Like now doctors and many other professionals are telling, many of the diseases are not really because of the problem in the body, but it is the percolation of the disturbance of the mind into the body. This is called psychosomatic principle. Now the reverse of this is also true. If the body is not immune, capable, enduring, then definitely the mind also gets depressed, anxious, sad. This is called a somopsychic principle, where the body influences the mind. So both type of stresses are possible. For example, when we get angry in our mind, but the blood pressure increases and blood pressure is in the body. So an anger in the mind increases the blood pressure in the body. This is a psychosomatic principle. But at the same time, suppose you take a very good head bath, cold water or whatever, hot water, whichever you like. You are taking bath to the body, but you will find immediately after taking bath, at least for a few minutes, you feel mentally a little relaxed and calm. The bath was taken for the body, but the mind became a little relaxed. This is where the body influenced the mind, which is called a somopsychic principle. So it is very obvious, if we have to reduce our stress, there are two ways of doing it. One, increase the capability of the body and make your mind to become very tranquil. In fact, that is the major reason why stresses are getting developed. If we take our ancestors, you will find that because there were no gadgets available at that time, they were doing most of the activities, household activities, other chores, routines, etc., by using the body manually. Therefore, the body used to get sufficient exercise. But unfortunately, because there is not so much of competition or so much of what I should say, zeal to achieve something great, the mind might not be extremely active, although it was active to some extent. Which means at that time, possibly, the body was hyper and the mind was a little hypo. Now, in the present days, exactly the reverse has taken place. Many times the body is very sedentary. People don't do exercise or yoga or gym or whatever it is. So the body is hypo. The metabolism is not okay. The resistance power is not right? But unfortunately, the mind is extremely active. People want to achieve a lot of things, want to make money, want to be in the right race, achieve as much as possible in the minimum possible time, overthinking, thinking on the same thing too many times, or thinking on too many things at the same time. So right now what has happened is the body is hypo, but the mind is hyper. So what is the solution? Very simple. Stimulate your body, tranquilize your mind, so that both of them operate in the same frequency, so that the stresses get managed. That is why typically any stress management program will contain two branches. One is to stimulate the body, the other is to tranquilize the mind. This is one way of looking at stress. So let me repeat. The stress happens because when we force our body and mind to exceed the threshold level. It can also be explained as the stimulation of the sympathetic part of our nervous system and not commensurate activating the parasympathetic nervous system. Stress can also be defined as the difference between the expectation and the reality. It can also be defined as a syndrome in which the body is sedentary and the mind is hyperactive. So this is the general interpretation of stress. There is another interpretation. This is based on the two halves of the brain. Many of us are aware we have got a left brain, we have got a right brain. The left brain is responsible for science, technology, data, statistics, derivations, quality, the materialistic science, for example, the structured way of thinking, the agenda-based discussions. All these things are a part of the left brain. The right brain is responsible for emotions, our feelings, creativity, values of life. These are the things which are there in the right brain. When these two brains are not properly balanced, in the sense we do not know which brain to use under which situation, stresses will get developed. So therefore, we need to stimulate both parts of the brain equitably, may not be equally, and use that part of the brain which is demanded by the situation. Then you will not be under stress. Let me quote an example. For example, let us say that right now I am talking to you, there is a laptop, and let us say laptop crashes. And due to some reason, it has stopped. So now I need to stimulate my left brain to think logically. Why did it happen? Was there a battery problem? Is there a virus? Is there a technical issue? So I need to think logically from the left brain. Suppose on the other hand, I feel very guilty about it. I feel very sad or bad about it. I feel very much irritated because of that. Because I'm using the right brain, I'm under stress. Same thing happens when we are required to use the right brain. When we use left brain, also stresses will get developed. Suppose you are going to see one of your friend who is in a serious condition in a hospital. You are supposed to look at him with a lot of empathy, with helping attitude, sympathy, or whatever. Suppose at that time you become a little commercial oriented and try to think what is in it for me. By stimulating the left brain, we are under stress. So these are the various nuances of the stress. And before I get on to the topic of management of stress, let me put some slides to authenticate whatever I have told till now. Naveen, you can start putting the slides. I will also suggest let us make the program very interactive. So any doubts, any clarifications you want, put a question in the chat box, and Mr. Naveen or someone there will tell me, and I'll try to answer to the best of my capability. So as we told, stress is the result of a person being pushed beyond the limits of his natural capacity. It is also the non-specific response of body or mind to any demand made on that. It is also the arousal of the sympathetic branch of our nervous system. Next. As I told, the mind influences the body, which is called psychosomatic, and the body influences the mind, somopsychic. So the body and mind should be synchronized to ensure the stresses are kept at the barest minimum. Next. As I told you, the example of if the violin strings are very loose, you cannot practice good music. So very less stress makes our life lackluster and no enthusiasm. Similarly, if the violin strings are very tight, also we can't get music. Like that, if we make our stresses to go beyond a limit, we will break down. If the violin strings are optimally stressed, tight, then we get the melodious music. So we need to optimally stress ourselves. Next. This is what the right brain and left brain syndrome which I was talking. The left brain is responsible for thinking, intelligence quotient, technical skills, analysis, logical data and statistics, science, management, science-oriented, success-oriented, commercial sense and competence. Whereas the right brain is generally responsible for feelings and emotions, emotional quotient, human skills, relationship orientation, creativity, intuition, artistic pursuits like singing, dancing, sculpting, then leadership, satisfaction oriented, higher values, and character. In other words, left brain is the chemistry of life and the right brain is the poetry of life. Now, when it is shifted to the teaching profession, the same concept of the model of left brain and right brain, it looks something like this. When we utilize the left, when we teach strictly as per syllabus, these are all the left brain activities. Maintaining timings and discipline, analyzing why a student can't study, data, statistics and students' marks, etc. Announcing the agenda of the class before teaching, teaching in a very structured manner like introduction, teaching and summarizing, disciplining the mischievous students if any, distributing the portions throughout the academic time equally, being firm and strict, writing organizedly on the board, sequential teaching, upgrading our teaching skills, giving clear notes, inculcating scientific temper in teaching. All these aspects emanate from the left brain of a teacher. But this is not enough. If we have to manage the stress, we need to use the right brain. And which are the right brain functions in teaching? Let us have a look at it. Care and concern for the students, empathizing with the students, that means having a lot of patience, smiling, a little bit of fun, creativity in teaching through games, pictures, cartoons, practicals, demos, stories, good relations with students, parents, co-teachers, superiors, managing the negative emotions like anger, professional jealousy, frustrations, developing patience and perseverance, flexibility in teaching methodology, participation of the students in the class itself, feeling proud about the nobility of teaching profession, making the students to share their experiences so that there is some cross learning if required, and developing higher values of life like honesty, integrity, gratitude, respect, patriotism, humility with the students. So if these two can be balanced and if a teacher can use the left brain when it is needed and the right brain when it is needed, his stress levels and thereby the stress level of the students can be at the lowest level. Okay. Yeah, next. As I told you, the stress can also be explained as the difference between the expectation and the reality. And there are two ways of doing it, which I explained to reduce the stress levels. Yeah, next. Clarity in mind and purity in heart is the surety for success. It is an excellent formula for stress management. If the mind is clear about the thoughts and there are no confusions, there are no lack of clarity, and the heart is pure with positive emotions like love, affection, respect, empathy, sympathy, transparency, honesty, integrity, a lot of stresses can be managed with these two syndromes of clarity in mind and purity in heart. Next. Okay, now at this point of time, next I'll be taking on how to manage our stresses. But I would like to invoke one or two small questions from the participants before I get on to the real management of stress. You can put your questions in the chat box, one or two, so that the session can be made a little more interactive. You can also comment. You can also comment on the session. How is it going? Is it fast, slow, okay? Are there any mid-course corrections which are needed? Feel free.
Good afternoon, delegates. Please give you the raise hand option so that I'll be able to unmute you. Yeah. You can interact with the speaker directly.
Yeah, only one or two so that we can proceed further.
We are supposed to get relieved of stress. Don't feel stressed to raise the hand.
Yes, yes, feel free.
Anyone?
Anyone?
How are you feeling attending this FDP for the 2 days on artificial intelligence, something which at least presently I'm learning about? Stressful, not stressed, easy? Anybody from the audience?
No, but the point is if people don't participate, initially for a few minutes, the audience will listen more out of interest on the topic or a little respect to the trainer. But afterwards, they listen more out of sympathy to the trainer. Anyway, I think the trainer himself is enjoying way to get into problem. To ensure that syndrome does not set in, I just invoke one or two questions. Feel free to ask.
Sir, I have a question.
Yes.
The type of stress during pandemic.
Hello? Hello? Yes? Yes.
Sir, I have a question here. The type of stress normally people felt during pandemic. Share some ideas on that one.
Sure, sure, sure. See, pandemic, the biggest problem of the pandemic was the uncertainty. See, there is what is known as a foreseeable stress. For example, let us say that tomorrow you have to go for an interview or you have to make a world-class presentation, and today you are making preparation. You can imagine that tomorrow 10:30 you have to make a presentation. So in a way, you are getting planned to manage that stress. That will not be that much stressful. But unfortunately, pandemic has taken people by surprise. It has exploded. It has come all of a sudden. And that has created tremendous amount of stress in the people because they were not prepared. Now, what is the problem pandemic has brought in? One is, of course, lot of fear, especially the first wave, and more important is the second wave. That will I contract the COVID, for example, and lot of information we were getting, some of which was contradictory also, and there was no solution in sight in respect of discovering a what I should say vaccination. So it was highly stressful at that time, and it also put people into a lot of financial difficulties because the savings started getting melted out, and a lot of industrialists were finding it very difficult to cope up with the situation because they were not geared up. There were no enough reserves, and it also shattered some of the deep rooted myths which people had. It made, in my opinion, it made people less materialistic and more philosophical and spiritual. We came to know due to pandemic, in my opinion, that matter doesn't matter. Matter means your houses, your wealth, your money, your gadgets, your possessions. But only no matter matters. No matters like peace of mind, relationship with the spouse and children, your emotional stability. So pandemic taught us that matter doesn't matter and only no matter matters. And many of them were not prepared for these things. For example, this word called as mind or mental health was not much used before pandemic. But you see, after the pandemic has come, you see the number of times this word mental health or mind or how to be peaceful. Almost every day you see articles, you see magazines, you see Facebook, anywhere that is being pervasively used. So, pandemic has been a very disastrous on the humanity, but it also taught us some lessons, but in a very, very painful way. Now, of course, people are coming to the terms and things are becoming better. And that is one of the major situations, the anxiety, the uncertainty, the fear, psychosis, the depletion of the finances, for example, and being at home, unable to move out. You see, and especially the people who are always outward flowing, always on the move, locked up in home, created a number of physical stress, mental stress, intellectual stress, emotional stress, social stress, and all sorts of stresses. So slowly people started understanding, coming to terms with, and I think things are getting stabilized now. And that is how this I would interpret. In fact, from the medication, people started moving towards meditation. From the capability in profession, people started believing in capability of stress and management. instead of the rat race in the profession, people started thinking that peace of mind and emotional stability are more important. We came to know the impermanence and the futility of all materialistic things and the need to become more value oriented, more philosophical oriented, and even spiritual oriented. We came to understand that we need to respect the animals, the plants, and we need to believe in an inclusive society, not man exercising his authority over all sorts of things. So I think these were some of the stresses which got generated during the pandemic, and we are trying to overcome them. And in the process, we learned a few lessons also. This is my interpretation of the pandemic stress. Am I clear, sir? Who asked the question?
You are clear, sir. I have one more question right
Please.
back. How
Please.
best we need to take up life post-pandemic? Lessons learned.
Yeah, we need to, number one, have a very clear lifestyle, because we understood that the most important thing which increases our immunity power, which bestows peace of mind, is to have a good lifestyle. Now, this lifestyle, I have created a module for that. Let me explain that to you. One, we need to take care of the physical aspect of our personality, which only means we need to take balanced, nutritious food. We need to drink sufficient quantum of water, do physical exercise every day, keep smiling in spite of all the difficulties, have correct postures in sitting or standing or walking, be in sunlight so that you get absorbed with the vitamin D, have regular health checkups, learn to rest and relax, and get a blissful and deep sleep. All these things are pertaining to your body. And many of you agree that some of the people had ignored a lot of these things. Physical exercise were not being done, they were not sleeping properly, and no sunlight, for example, postures were inappropriate. So, if in the post pandemic, any time for that matter, not only post pandemic, we need to take care of these habits from the physical point of view. Sedentary life is one of the worst possible things. Our body is not meant to be in one place, it is meant to move. I always say movement is medicine, sedentary is sickness. MMSS, I call it as. In fact, I am now 72 years old. From the last 60 years, that is when I was about 10 years old boy till today, I get up at 3:45 in the morning every day. And every day 1 to 1.5 hours of little bit walking, little bit of yoga, little bit slow breathing. I definitely do almost every day, minimum 6 days in a week, if not 7 days. Either you use your body or you lose your body. So long as your physical existence, you should do physical exercise. Anything including your body, if it is not used, it becomes useless. The second thing is food. There are 3 types of food: tamasic, rajasic, and sattvic. Tamasic foods make you very inert, lack of enthusiasm, sedate, lazy, like toasted food, roasted food, oily foods, bakery products, etc. The rajasic food will make you stimulate your sympathetic part of your nervous system and make you very aggressive, angry, jealous, all sorts of things. Like spices, for example, high level of anything which in moderation is perfectly all right. Sattvic food is that which creates an equanimity in your mind, endurance in your body, like fruits, vegetables, fibers, a little carbohydrates, little proteins. So we need to balance out these things. So I think there's a very great need to take care of the body. The second thing is to develop a good mental ambience by developing a positive attitude, optimism. Very nicely it is told, it is not only your aptitude, it is your attitude which decides your altitude. We should think positive, that yes, everything will go on. This too shall pass off. After all, humanity has seen a number of pandemics, number of epidemics, number of endemics. This is one of. Maybe it was a little more pervasive and a little more uncertain. We should always think very, very positively. And we should develop book reading to enhance our knowledge, our skills, so that we feel ourselves contemporary. We need to play puzzles and riddles so that our neuropeptides, neurodendrites, and the whole lot of synapses get formed in our brain, and our brain will be always alert, agile, vigilant, and ready for action. We need to develop creativity, thinking out of the box, lateral thinking, divergent thinking. And therefore, we need to develop new skills, avoid criticism on others, take ownership, and develop a sense of gratitude for the things, for the people who helped us. Introspect ourselves very plainly, look into yourself, and sometimes in a day, give a mental holiday, make your mind zero thoughts. No, these are all the activities you need to do to ensure your mental peace, which is very much needed. As I told you many times, it is the mental disturbance which manifests as a physical disorder. The number 3, more important, is the emotional aspects. Spend more time with your family, the quality time with your spouse, children, in-laws, parents. A domestic disharmony can lead to a corporate calamity. There is no point in being an intellectual giant in the profession and an emotional pygmy at home. There is no point in being extremely popular to the world outside and being a stranger to your own people at home. So we need to give family the due importance. And then prayer, an excellent psycho-emotive therapy. Listening to melodious music, which makes your brain to get into alpha state. Looking at nature, its splendor, its grandeur, the colors, the birds, the flowers, the trees, the sky, the moon, the mountains. Developing a sense of appreciation for whatever is good in this life. And having good relationship with friends. And being choosy with our friends, because our friends determine a lot of personality characteristic of ours. And serving humanity. And if possible, being with pets. All these things will take care of your emotional stability. Your emotions will be stable and they will be noble. We will not have negative emotions much like anger, jealousy, greed, lust, aberrations, inferiority complex, fear. And we also need to develop positive hobbies like singing, dancing, reading, so etc. And reduce our dependence on the gadgets like TVs, mobiles. And avoid gossips. Don't talk anything which doesn't pertain to you. So much of trolling, so much of criticism goes on. It is sickening. And don't get into any bad habits like smoking or alcohol or anything else. So this is the entire package. Post-pandemic and even forever, we need to develop a good immune body, a peaceful mind, a stable emotions, a sharpened intellect, and a overall holistic personality. I hope this is clear. Any questions on this?
So it's very clear, sir. Thank you very much for your wonderful thoughts, sir.
In fact, what I have done is I have structured, because many times we listen, we don't practice.
Yes.
An ounce of practice is better than tons of theory.
Right.
So what I have done, I have created a chart, and I have been following it from the last 50 years. All these activities I have written down, and every day we need to tick which activities we did, which we did not do, so that there is an accountability for our lifestyles. I'll just show that chart to you. It's now in timetable. Slide number 8. So you can see here what I have done, sir. You can see date. Actually, there must be 7 columns because it is 1 week per sheet. Physical activities you can see here. Suppose you did some physical activities, walking, a little bit gym, aerobics. On that date, you have to tick it. Did you get enough sleep? Was the sleep very deep sleep? What are the type of food consumed? Suppose you ate a lot of wrong food. Then you have to write not good, NG, under 3A. Suppose you ate too much, for example, which is more than what is needed. Then you have to write not good. Did you get physical rest and relaxation? Suppose it was right, put G. Did you stand under sun at least for 10 minutes in the day? Tick it. Did you at least consume about 8 glasses or about 3 liters of water? Tick it. Like this, I have created an entire stress management and positive lifestyles. Now see the mental activities. These are all coming under physical, connected to body. Next, when you go to the mental intellectual, we need to develop slow breathing and meditation to make our mind very calm. Reading educative books to enhance our knowledge. Action taken to our professional skills. So a lot of new skills are coming even in teaching itself. We need to develop those. We need to reskill, upskill, and multi-skill ourselves. We need to play puzzles, riddles to stimulate our brain. Creative thinking, innovation, positive self-talk, what we talk within ourselves. Introspection at the end of the day, before you go to bed and sleep, just have a flashback of whatever we did during that day. Whom did we help? Whom did we hurt? What did I do wrong? Where did I gossip? What value addition I have done for others? Avoid criticism on others. Develop some hobbies. So these are all coming under the mental intellectual. Then we have the emotional aspects, like prayer, listening to melodious music, watching nature, time spent with spouse and children. Number of people who are appreciated. See, I have taken a target. Without appreciating 3 people in a day, I will not take dinner in the night. So what happens now? Because you need to take dinner, you start searching for good things. I'll tell Mr. Sajid sir that your watch is good. So one is ticked. See, I always say good practices you need to force on yourself. Bad things will come by itself. Good practices are difficult to start, easy to quit. Say tomorrow you can start yoga, difficult to start. But after 1 week you can quit. Bad habits are easy to start but difficult to quit. Suppose alcoholism, very easily you can start off. But it may stay with some people as an addiction. So there is a need to document. There is a need to put it in a format so that we are reminded every day what we need to do. Being cheerful, smiling. I always say instead of worrying and having a wrinkle on your face, why don't you smile and have a dimple on your face? Smile, it increases your face value. Your dress is never complete unless you wear a smile on your face. People tell me, sir, good days are not come. Therefore, I can't smile. I tell the other way, it is not like that. You smile without reason, good days will come automatically. Then the social aspect, like doing social work, helping others, interaction with friends and relatives, attending social events. So when you come to the lifestyle, you can go to the next slide. Anytime you do a job which you are postponing for a long time, you feel extremely happy actually. Your stresses will go off. So did you do any job which you are postponing? Any new skills you learned? What are the quality of TV programs you watched? Were they interesting? Were they useful? Were they educative? Or were they more violence and horrible things? See, it is a garbage in, garbage out. Whatever you see and whatever you listen, that is how the thoughts will come out. It all depends on what you're feeding. How did you optimally use mobile phones, social sites, selfies? And finally, did you avoid gossiping? So this is the chart which I have prepared for stress management lifestyles so that there is an accountability and there is a documentation so that they don't skip our notice. Any questions on this? Many people feel I can't do so many things. Actually speaking, we need not do anything more. See here, what I have done is, all that it is needed is a restructuring of your timetable. Not 1 minute extra I am asking. No, not needed. Suppose you avoid over usage of your mobile and you can save 1 hour. That 1 hour can be used for a little physical exercise, little nature watching. It only needs a rearrangement, reorientation, refabrication of your lifestyles. No extra time is needed generally. So this is where I have explained. These are the extra time needed activities. This is where you save and they balance out almost. This is what is very important to manage our stress. Now I invoke further one or two small questions.
Sir, good afternoon.
Good afternoon. Yeah.
Yeah, it's not about me. Like you were talking about rescheduling, right, sir? It's about
Yes.
my daughter actually.
Yes, yes.
She reschedules very nicely. Every day she makes a fresh timetable. When it comes to implementation, it is 0 and we have a fight every night.
Yes, yes, yes.
Nothing has been achieved that day.
That is the problem. So there why does it happen? It
Not
happens with us also. Suddenly you listen to a motivational talk, you get inspired to do a lot of things. How long will it stay? Few days. Then we'll be back to square one. It's because, in my opinion, it happens because we have intellectually understood the concept, but we are not fully convinced about what we need to do. And we are not experientially authenticated. So for that, these type of activities, and you need to be a little harsh on yourself. Suppose you take a stand, 10 minutes if I don't walk, I will not take dinner. And you stand by your commitment. Possibly you will walk at least 5 minutes, you will walk. Because we are too loose on ourselves, too casual about approach. There is no moral fear. All these things will happen. The discipline is very, very important. If you take the word discipline and add all the letters, see, D is English alphabet 4, I in discipline, DISCIPLNE, I is 9. You total it up, it exactly comes to 100. You need to be 100% disciplined in life. So this is what we need to teach children. One, by role modeling ourselves, we need to follow that. Number two, explain to them if they continue to be like this, callous or casual, what will be the consequence of that? You take the most successful people, one common factor in them are two things. One, they are disciplined, whether it is in lifestyle or maintaining commitments or coming to in time to events or programs and meetings. Two, they constantly read. So we need to develop these activities. That's why I do a lot of programs for school students, college, at least about 100 colleges and students I keep teaching. The importance of discipline, the importance of having proper lifestyle, the significance of studying, for example, developing concentration, memory power enhancement, developing positive values of life, feeling proud to be in our country, whatever. And that's very important. Good. Any other questions? Any other? I just wanted to have a midterm opinion about the session. It is something like in the films they ask in the interval, how is the film? No, some one or two comments so that I know whether I'm making some value addition. Few comments from some of the participants.
Delegates, please participate and
Yes.
you can raise your hand. I can unmute all of you otherwise. We are not unmuting, sir, because some disturbances might come. We don't want to interrupt the session. That's why.
Sure. But they can put something in the chat box or they can raise and talk.
Yes.
One or two, I would like to listen or hear or in the chat box, one or two comments on about the program till now, whatever has happened. Because that gives me an idea.
This is a testing for you, sir. Not
I know. And they will write some of the, because without the comments program will not continue now.
Yes. So we have one from Sonali who's telling it's an excellent session.
Sure.
Then first half it's going good. So far from Dr. Venkatesh.
Nice to hear that. Any other comments?
I think dear delegates, just maybe like sir was telling now, we need to mobilize and motivate ourselves. Just one comment here. You can take a moment to do that. We have 3 more days to go with the FDP.
Yes.
The spirit can't be killed.
Participation is very, very important. I'm seeing here, first half is very nice. In the world of variations, how to keep so many things constant. Okay. Good session. Very informative and thought provoking. I have a question.
Dr. Divakar,
please, can you ask the question, sir? Delegates be interactive. Okay.
Dr. Divakar, would you like to unmute? Please raise your hand. I'll be able to unmute you.
Yeah,
Anybody,
please.
any of the delegates?
Make it as interactive as possible. It helps all of us. After 1.5 days of human AI is working wonderfully. Okay, let's clear that. Okay.
I think you're able to see that.
Sure. Explain good various types of stress. Yes. Nice. Okay, I think by and large it is going on fine. So it looks I can continue.
Yeah, Manoj, so how to inculcate this activity in our BTech students?
Yeah.
From Dr. Divakar here.
From Dr. Divakar, how to inculcate in BTech. One, whoever, there are two ways of doing it. Whoever does these things to some extent, say he's disciplined, he's studious, and he's having good values, appreciate them. And there is one rule, appreciate in public, criticize in private. Appreciate in the class itself. Of course, without hurting others. So everyone will feel, oh, I would also like to be appreciated. That is one way of doing it. Rewarding, for example. The other way of doing is giving them a chart like this. Like, for example, if I request all of you and force you have to fill up this chart, come what may. At least you will do out of the 25 activities, at least 5, 6 you will do. And after about 10 days, you will do 10 activities. So unless you try to measure any activity, see, there is a very good statement. It says, anything which cannot be documented cannot be understood. And anything which cannot be measured cannot be improved. So make a small document like this. What are the characteristics you want the students to develop? On what scale they are right now on a 0 to 10 scale? After 1 month, redo it and find out whether they are improving. So you need to create certain simple, very simplistic documents so that there's an accountability. There is another way of introducing BTech. Then give for example, some case studies. Take some examples of BTech students who have taken the studies seriously, their character seriously, and have reached very high positions in life, making a lot of money in good ways and becoming very popular. Give such examples without, of course, exaggerating or glorifying them. So by these activities of rewarding people who follow, giving them a format to document, giving examples or case studies of people, it is possible that to inculcate certain degree of these activities in the students. How the dynamic teaching and brain dominance are equally important in mentor-mentee program. Yeah, see, mentor-mentee, unlike the class, mentor-mentee is always a little long drawn. And in the class, you are more predominantly it's a knowledge transfer or skill enhancement. Whereas in a mentor-mentee, it is more of life skills, like how he handles relationship issues, how he has to become more, what I should say, dynamic, etc. So mentor and mentee relationship is a little more far long term as compared to class, because in the class you got a specific objective to do, finish the syllabus and make them examination friendly or examination certain. So mentor mentee will have to get into a different frame of work if you are really mentor. Although a teacher should also be a mentor to a certain extent in the class. Now, what are the clinical measures in stress management? Many of the bodily parameters will get affected. When you get stressed, it is told that your blood pressure increases, your cardiac beats will increase, your pulse rate increases. Sometimes you may have more acidity, for example, flatulence may increase, you feel irritable, you will find those things which were giving you pleasure earlier, today they are not giving you. You will not be able to undergo stress test like treadmill test, etc., which they do in clinical tests. So a lot of bodily parameters, when measured, can indicate whether you are under stress. But the best person to decide whether you are under stress is yourself. It is because the stress threshold level is very individualistic. See, suppose a student fails. 2 students fail. One boy may get extremely stressed. Other person may not get stressed and he may become more careful and more vigilant and more optimistic. Which only means that which stresses person A need not stress person B. So very clear clinical parameters is not possible in stress. We can very clinically measure your temperature, measure your blood pressure, measure your blood sugar levels. But that is not possible in the case of stress. We have to go only by the manifestations and the subject experiences of the personality. So that is how clinically they are measured. Although there are a lot of research going on, can we measure, for example, if your immunity power goes down, it is said to be you are under stress. If your parasympathetic system is overdrive, using too much, you are under stress. If you cannot peacefully sit in one place, you fidget too many times. These are some manifestations of stress. So like this, there are a lot of physical parameters also which people are attributing to stress. I need to call you for an offline session to manage stress post-pandemic in students. Most welcome. See, in the pandemic time itself, I have conducted about 300 webinars for different colleges, schools, industries, organizations, Rotary clubs, Lions, etc. So please, there is a need for these values to be disseminated. I'll be more pleased to do that. How conflict management is handled in institutions at upper level when subordinates are the sufferers. Fight, flight, and flow. If the institution is having some problem, maybe they are apathetic, or they are not rewarding, or there are too many heads in the top, whatever, the first thing you need to do is fight. Fight is not rebelling against. No. You please talk about it. I always say this world has got into problems not because of the wicked people. It is because of the silence of the good people. So you need to bring it to the notice in a very firm but polite way. Suppose it is not possible, fight. Then you flight. Flight means go to other department, go somewhere else. Suppose that is also not possible, then you flow. Flow means you compromise. Now, when you flow, you can flow with two things. Either you flow with pleasure or you flow with pain. When you are told that fighting is not possible and flighting is not possible, the best thing is to flow, but flow with pleasure. Don't flow with pain. I call this as a technique called 3F plus 1P. Fight, flight, flow. If you have to flow, flow with pleasure. If you flow with pain, the problem is with somebody else, but you are giving a lot of problems to your own body, your own mind. Why are you making your body to suffer, your mind to suffer for nobody, for none of your faults? So that's what you need to do if that is the situation with anybody, not only institution. With people, with your spouse, with your boss, with your neighbor, with your authority, whatever, or your subordinate. Fight, flight, flow. Flow with pleasure. How to manage and overcome the stress in busy academic audits like NAC, etc. Number one, proper planning. Very good prioritization. 3, don't get panicky. 4, simplification processes. 5, delegate with others and get the jobs done. 6, don't get overwhelmed. 7, use a Pareto principle. What is this Pareto principle? 80-20 it is called. 80% of your problems can be solved with 20% of the activities. So find out all the problems, catch the big fish and clear it first. So by properly managing your time, by planning, organizing, systematizing, simplifying, delegating, you can come out of these problems to a great extent, may not be full extent. Temporarily join the start. Are my what I should say, inputs useful? Madam, please ask the
Yes, sir.
participants.
Yes, sir. Very much. We are getting
Good.
positive feedback.
I think a lot of people are asking questions. They have started open up. That's what I want.
Threshold has been crossed, sir. So we have
Yes,
one question that you missed in the beginning from Mr. Sumit Joshi. In the world of variations, how to keep so many things constant?
See, variations are only in superficial things. Our value should be constant. This is a very important aspect. This world has got a good equation of stability and mobility. You cannot be mobile always. Something needs to be stable. So you need to be stable in your values, in your peace of mind, your emotional stability, in your relationship. But over that mobility is possible. You can change your food habits, dress, your interpretations, etc. So always get yourself connected to something which is stable. Then you will not get overwhelmed with the things which are changing. It is. I always give an example of the watch. You know, in the watch, the needle is moving. It is mobility. But imagine that the dial and the numbers also start moving. How will you see the time? Numbers and dial needs to be stable. On a road, the car and the trucks are moving, agreed. But the roads needs to be stable. Imagine the road also starts moving. What will happen? So this mobility for the sake of mobility, change for just the heck of change, is a very bad syndrome. You need to change for better. So don't unnecessarily give your mind and body to too many changes. You need to adapt to changes, I agree, but you should not become obsessive. Obsession to keep on changing, then you will never have peace of mind. So this is how you need to find out what are those stable factors in the everlasting, ever-changing world, and hook on your mind and your intellect and your emotions to those standard and stable things. Then you will not get that way. For example, body is unstable, but your spirit is stable. For example. So these are the philosophical and spiritual approaches which you need to take to remain stable in the chaotic, ever-changing world and universe. Good. Anything else, ma'am? How to manage unachievable tasks in a short period which create a lot of stress at work. Now, if you have decided unachievable, see, don't mistake me. Many things which people thought unachievable have become achievable today. There is a very nice statement which says, impossible is an opinion, it is not a reality. Sometimes it is a reality. So don't start with a paradigm that is unachievable. It must be achievable. Maybe it takes longer time. It puts in a lot of efforts. You may need a lot of help from others. You may need a change in your attitude or values, whatever. So, number one. Number two, if you are convinced that in 2 months, this is the maximum a human being could do, why are you bothered whether it is achievable or not achievable? That is, see, there is a very beautiful system. It is called process happiness versus destination happiness. You got in 2 months, you have to achieve something. Don't always think of I have to achieve. It creates a lot of stress. But enjoy the process on the journey of achieving. You will not get that much stress. And even if you don't achieve a part of what you would have achieved, you will not feel guilt and you will not feel bad because you have done your best. See, however good program I do, few people among the audience will say program was not effective. Fine, it's okay. Let me take with a pinch of salt. Let me learn from them. So your obsession that I want to complete everything in this rat race, that's what puts a lot of stress. So when an unachievable target is there, number one, take out from your mind it is unachievable. 2, work at the maximum capacity and achieve whatever is possible. And don't feel guilty or don't feel bad about that part which was not achievable because it was just not achievable. Okay. Something. Okay, wonderful. Awesome session. There is unachievable. Let me take this. Well answered, Sajeev sir. Okay, fine. Fine. Any more questions? Okay. Let me talk a few minutes on yoga before how to deal with frustration if the end result is not as desired. What is a better choice? First of all, your passion for end result should go, sir. Result will not justify the means many times. Take your own profession. You say the end result of a student must be he should get more than 90 marks. Agreed. Imagine he gets 90 marks, but very illegal methods. Will you still justify the end result is achieved? By all wrong things, by what immoral, unethical ways? All that I am trying to tell you is end result is only for guidance. It is no authentication, it is no justification for how much efforts you have put in and what is the process you have undergone. So forget about, see, what does our philosophy has taught? Karmanyeva, what do they say? Keep on doing your job with the best possible intentions, highest possible efforts, greatest possible commitment, with totally unflinching dedication, without any ego. Forget the results. Results will come. And if it doesn't come, it will come in another form, in some other day. So this passion for the end result should go off, sir, actually. Whether it is in education or in industry or in social life. That is why I always tell in my program at the end, I'll tell, I don't mind if you are an ineffective professional. No problem. I can teach you the skills, but don't become a bad human being. That is a problematic. Values are more important than tangible results. So don't get too obsessed with results. What is the better choice? The better choice is to enjoy what you are doing. As I told you about 5 points. Number 1, your intention should be very noble. Anything what you do, put a question. Why am I doing this job? Why am I doing this program today? Is it to get more popularity for me? Is it to get more name and fame? Is it to get your appreciation? No. It is to tell to some of my friends something little what I know. If it can add their value and they can get some insights, I am blessed. If that is the intention, you will find everything what you do becomes a yagna. Yoga karmasu kaushalam. The skill in the work is called yoga. So let us come out of this syndrome of end results. Let's enjoy what. That's why please don't tell your students work hard, study hard. Don't say. Study joyfully, study enjoyably, study responsibly. Marks will come. Madam, ask people whether I'm making some sense or no. You have to moderate in between.
You are, sir. Yes. Messages coming. We have a WhatsApp group also. There also people are responding in a positive, happier way.
So this undue tension, undue this, I don't know who taught. Everything is decided by your end result. Who told? For example, there are many certificates, especially in the engineering industry and even pharmaceuticals. They don't check the end product at all. They check the process only. Because they say if the process is good, the product will definitely be good. So please, if for you also, your children, your students, please don't focus exclusively on end results. Yeah, it's very much needed because see, process cannot be measured many times, end result can be measured. That is the only difference. To that extent only we give importance to end result. How you made money is more important than how much you made money. See, these are the good values which we need to give it to the people. Okay, path is also important. Not path is also important. Path is more important. Sumit sir, are you there?
So you are like a teacher now.
Path is also important means as though result is important. Path is no. Path is more important than result. Tell your children like that. See, the people who are sitting here will decide the destiny of this country because each one of you are disseminating knowledge, information, skill, values to your people. If a country has to prosper, only one set of people can do it is the teachers. I am convinced beyond doubt. That is why after working in an industry for 20 years, I undertook teaching. In an industry, I was having a very good career, but I thought, no, this is the noblest of professions. The flowers blossoming in front of you, the personality oozing out with enthusiasm, adding value. So there is the responsibility we need to feel with the children, with the students, sorry. Very much needed session. Thank you. And judge you on the basis of just, yeah, I agree. I agree that judgment is done based on that, but that should not be your focus. Because see, if the judgment is not on the end product or the end result, on what can I judge? No, I need some parameter to judge. Agreed. But that does not justify that the means was okay. Agreed. Any other questions? Any other doubts? See, ultimately, what is needed, let me tell you. In yoga, also, what we talk. One, listen to me carefully. Or I will tell you physical immunity, the most important thing. Your immunity power. Two, mental tranquility, peaceful mind. Three, intellectual clarity, correct decision making. Four, emotional stability, stable emotions without mood swings. Five, enhanced creativity, thinking of new things out of the box. Concentration intensity, ability to instantly concentrate on a particular thing without any distractions. Material prosperity, making a lot of wealth in a good way. Money is very important. And material prosperity is very important. Social empathy, developing a service attitude and vibing with the difficulties of others. Domestic unity, unity with spouse, children, etc. And finally, spiritual identity, realizing our ultimate existence, supra consciousness, and getting aligned to that cosmic will. So, your equation should be physical immunity, mental tranquility, intellectual clarity, emotional stability, concentration intensity, enhanced creativity, social empathy, material prosperity, domestic unity, and spiritual identity. That is the menu for your development, stress management, and that's what yoga teaches. Naveen, you can put that slide. Some of you can send the slide also. Mr. Sajeev sir, I will send some of these slides. You can share with the people.
Sure, sir.
Yeah. Naveen, put that slide physically. I'll tell you where it is. Go to yoga, yeah. This one, yeah. So you can see this menu, what yoga tells you. Put the whole thing, physical immunity, mental tranquility, memory longevity, concentration intensity, increased creativity, intellectual clarity, emotional stability, social empathy, domestic unity, relaxed prosperity, and spiritual identity. Please take a copy of this and fix in your rooms, walls, in your schools, colleges. That should become the guiding factor for all of you. And that's what yoga is all about. Yoga works on all these things. It makes you physically strong. Mentally, you will be very peaceful. Your memory power will increase. You will be able to concentrate very nicely. Your right brain will get developed and therefore your creative instincts will improve. You will be able to decide very, very correctly, right decisions at the right time from the intellectual point. You will not be given to bad emotions like anger, jealousy, frustration, greed, lust, all nonsense. You will be able to vibe with other difficulties. You will have a wonderful family life. You will be materially also prosperous because many of these things are required for making money and becoming more wealthy. And above all, you will realize your spiritual connotation and spiritual connection and spiritual identity. These are the benefits of yoga. And for that, we do a lot of activities like yogasanas, slow breathing, meditation, withdrawal of senses called pratyahara, yama and niyama, which are moralistic and ethical principles, dharana, which is called concentration, dhyana, which is meditation, samadhi, which is communion. So yoga gives these 8 steps for achieving all these things which I've been talking to. How not to be influenced by toxic people despite being with them in the work area. Dealing them mentally, be with them physically. You have seen a lotus flower and that there is a drop of water. Will the drop of water mingle with the lotus flower? No. You just do like this, it drops off. But is the drop of water away from the lotus flower? No, it is sticking. So we with the fellows, but mentally divorced. Because if you want to avoid all toxic people, you will become a loner in this world. But don't get influenced. See, if you decide not to get influenced, nobody in this world can influence you. So this type of detachment in attachment and attachment in detachment need to be learned. Be with people, but don't get attached with them. That's how you avoid the toxic people. How to get rid of inferiority complex? One, enhance your knowledge. More knowledgeable you are, less inferior complex you will feel. Two, walk straight. Don't walk with a hunched back, which will give more inferiority complex. Walk straight, not very arrogant either. Three, raise your voice pitch by about 20%. Inferiority complex peoples talk at a little feeble, low tone. Raise your voice. Number four, do yogasanas in which the chest will be stretched backwards, by which you will develop as you are ready to face all the challenges. Like Ustrasana, for example, Bhujangasana. These are extremely good for developing the assertiveness or courage. Number five, ensure that you have a very good lifestyle. Last, very important point, take care of your friends. Very nicely it is told, tell me who are your friends, I will tell you who you are. Your friends will decide what type of personality you are. So don't be in the company of pessimistic, disempowering, horrible, negative, all sorts of nonsense they are talking. Most probably that's what we'll imbibe. Be in the company of pragmatic, optimistic, futuristic, intelligent, ethical, moralistic people, then you will develop more courage or more right values. So these are the ways in which you can come out of the inferiority complex. Delegates free to connect with sir and share your concerns too. Get clarity. Good. What else? Questions? Shoot questions. Shoot questions. I like questions. Are you getting the right insights, if not solutions for your questions?
Yes, sir.
Madam,
Yeah,
ask the people.
we are getting the right answers.
Good. Shall I, Sajeev sir?
Yes.
I have made a small note
Yes.
on the stress of exclusively teaching community. Do you think it will be useful?
Yes, it will be useful, sir.
Earlier. Now
Yes.
in put 21. I have made a small note of
Definitely.
what are the problems, stressful situations, or the challenges of teaching people. Very quickly I'll go through because I think time is running out. What are the typical problems which teachers undergo? 21. See, first is standing for a long hours. So what is that you need to do? Practice Sarvangasana, where your leg will go up and head will go down. There will be a better flow of blood to the brain. And there will be a relief to your legs. Before going to bed, keep your feet in cold water. Practice suggestive relaxation to legs. See, I've written a lot of books and made a lot of CDs on stress management, yoga, others. You can take that and practice those things. Speaking for a long time is another occupational hazard. So what you do? Gargle throat with hot water and salt. Reduce taking cold items. Practice pranava pranayama. It is a combination of akar, ukar, and makar, touching all aspects of your vocal cords. If you do that exercise, your voice quality will be very nice. See, I've been talking like this from last 28 years. Almost 25 to 26 days in a month, I'll conduct programs, morning to evenings, including Sundays. One of the reason, I'm not talking about myself, is because of these practices. Next. Not at par with latest subjects chapters. See, things are changing, so you need to be very contemporary. Update your subject knowledge constantly by reading, browsing internet, attending seminars. Very nicely it is told, if you want to go back to in the world, you need not walk back now. You stay wherever you are. All others will go in front of you. You will be automatically at the back. So you need to be very updated. What is the latest teaching skills? What is internet doing? How online sessions are conducted? For example, how simulation exercises are done. case studies, role plays, group discussions. Now, younger generation students not respecting the teachers is a very big challenge to the teachers. Number one, simple, don't expect the respect. Consider teaching as your duty and responsibility and be done with that. Because you expect only, as I told you, and they don't respect, that difference is the stress, no. So reduce your expectations. You are doing your job. Be done with that. Understand the immaturity, impulsiveness, and mischief liking of a few younger generation students. There is a characteristic. It is not that we were excellent students when we were young. Please remember. We were all reasonably well behaved because we did not know how to behave like this, and there were controls and checks and balances to control us. So love the younger generation. Don't become apathetic or don't become antagonistic to them. It doesn't help. So let me tell you one thing. You cannot change any person by hate. You can only change by love. Now, go to the next one. Colleagues rising up the position. You know, one teacher joined only 2 years back. He's already a professor or something. I am not. Fine. Don't feel jealous. On the other hand, learn from him. What is that competencies which he has developed? What are the skills which he has developed which I am lacking possibly? Unless it is a bad way of what I would say illegal and immoral, that we don't want in any case. Monotony in teaching profession. Introduce new techniques in teaching. Be creative and innovative. Experiment different ways of teaching. These are very important points. Realize that the subject and you are the same, but students are different every time. So every time you enter into the class, you are seeing a new set of people. So develop that thing. You know, I was talking to some hospital, I think. So one of the staff told me, sir, early in the morning at 9:30, if the patients come, we'll be a lot of enthusiastic. We serve them, we take care of them. But if they come at 5:30, we are also exhausted. We'll become casual. I told madam, what you say is right. For you, that patient may be 520th patient, agreed. But for the patient, that is the first time he's coming to your hospital. Please look from his angle. He has not come 520 times to your hospital. You have seen 520 patients, agreed. But when you look from his angle, that is the first time he has come to your hospital on that day. So you need to have the same enthusiasm, same care, same dedication, same commitment to this man who drops in at 5:30 as much as when he drops at 9:30 in the morning. Same thing happens with the teachers also. Subject you are told 1,000 times, agreed. Everything you know. But the boys are not same, girls are not same. It's a different setting. There is a lot of things which we can learn from students also, especially the present people. They are very well informed. Some of them are very focused. Introduce little humor, little anecdotes, practical exercise if possible, within limits. A good teacher is one who can make a familiar subject to look new and a new subject to look familiar. Any difficult subject you can talk in the day-to-day instances and make it extremely familiar. Anything which they think they are good, you can make it to look extremely new and very, very innovative and very different approach. That is a skill of a teacher. Next. These are all the challenges of teachers which create stress in them and you need to manage them. Next. Don't expect gratitude, not needed. Feel proud of such students. If see, many times what happens, your students outgrow in all aspects as compared to you. In money, wealth, position, popularity, fine. It is something to feel proud. It is not to feel bad about me. Now, sometimes we have spare time. Develop reading habits, develop useful hobbies like walking, yoga, gardening, meditation, social service, writing articles, books, music. I've written about 10 books. See, I've trained about 500,000, 5 lakhs of people I've trained. And I talk on about 85 topics in 5 languages, Kannada, Telugu, Tamil, English, Hindi. I got about 50 dancers who make dance dramas on educative topics. So you can do any amount of jobs. You should be a mountain of strength, a fountain of energy, and a volcano of enthusiasm. That's what a teacher should be. Next. Now, uneven workload, see, especially in teaching profession, uneven workload. Suddenly corrections will come, suddenly some knack will come, suddenly, and then there will be a little lull. So what you need to do, you need to plan, be proactive. When you have a little spare time, do some preparatory work. See, when you go to a hotel, whether you order for paneer butter masala, paneer matar masala, he will give in 15 minutes. How does he give? Because paneer is ready, the masala is ready, matar is ready. If you order this, he will take, this he will take, this he will take, do some heat it and give it to you. Many of such activities can be done in teaching also. Keep them in semi-finished preparatory work. Synchronize your lifestyle with respect to your workload. Suppose workload is very high, cut down on other activities. Workload is less, increase your other things like reading habits or being with friends usefully. Luring by peer, don't accept any favors from anybody for that matter. From students, favors, no obligation. Have high professional ethics. I always say a corrupt person has got a price. A value-oriented person, a good person has got a value. We need to be value-oriented, not price. Lack of physical activity, therefore walk around, stretch during rest hours, do yogasanas, aerobics, walk frequently, climb steps instead of using what I should say, staircase. Like that, you need to sandwich a little activity in your day-to-day. Slow breathing a little bit, make your exhalation twice the time as inhalation. Suppose you have to walk from your place to your principal's place. It takes 10 minutes. For 3 steps you inhale, for 6 steps you exhale. That itself becomes slow breathing techniques. So like that you need to sandwich physical exercise and breathing techniques into your day-to-day work. Okay.
Sorry to interrupt.
Yes.
We have another 2 minutes time, sir. We have few questions also in line to close the session.
Yeah, this is the last slide. Naveen, put the last slide. Yes, a teacher will never become obsolete if he updates himself. This is the profession where as age progresses, your value increases and there is going to be everlasting demand for good teachers. So these are the stress inducing in a teaching profession. Good. So with this, I am almost coming to a close unless you have any questions, any opinions, please feel free. Any thoughts?
So we have one question which is there in the chat box.
Yeah.
How to enhance team spirit?
Yeah, one is reduce the ego in us, look things from others' point of reference, empathize with them. Don't feel bad if somebody opposes your version. Maybe there is a great learning. In fact, in our life, we have learned more from people who oppose us, not with people who agree with us. Be clear about the end point that we are all working toward the same objective. Therefore, people can have different opinions, but the objective is same. It is not necessary that everyone should think alike. I tell in my programs, if everyone think alike, it means nobody thinks. Respect the different opinion from others. Don't get into jealousy, professional jealousy. Don't have groupism, either on language or on community or on religion or on the place of your native place, etc. Work towards collective working. Each person is bestowed with different types of capabilities. Some are good in analytical skills, some are good in administration, third person in accounting, fourth person in creativity, fifth person presentations. Let us synergize all this and produce a wonderful end product. Maybe a syllabus formation or a courseware or an event in the college. So by developing all this, by respecting others' opinion, being on the dot for the objective, liking the deviations, the variations from people, learning from others, not feeling jealous, not outbursting on people, controlling your emotions like anger, you see, developing helping attitude, synergizing the various skills, you can develop your team spirit. Fine. Any other things?
We don't have much questions. If anything
Sure.
is there, connect by WhatsApp, sir. We would like to
Yeah.
close this session, sir.
Sure.
Right.
So,
Sir, would you like to close the stress management session with a last word?
yeah. So basically, the main point is whatever I showed the slides to you, you should become a patana in your eyes. Whatever words I told you should become a shravana in your ears. This visual patana and the auditory shravana should get into your mind, called as manana. You should think that you should send it to your intellect where you do some chintana, and that you should put into practice, which is called chetana, till it becomes a part of your life called jiva. So I think let us put into practice.
A very warm welcome everyone to the valedictory program of this 5-day online ATAL sponsored FDP on artificial intelligence. We welcome our honorable chief guest, Dr. Manish Paul, campus director, Acharya, our respected principal, Acharya and B.M. Reddy College of Pharmacy, Dr. Amit Kumar Das, all the delegates and my dear colleagues for this valedictory program. It gives me immense satisfaction to share with you that we have successfully completed 15 scientific sessions with renowned speakers from all across India and abroad across these 5 days with the support from everyone. I now request our principal, Dr. Amit sir, to address the gathering. Sir, please switch on your camera.
Is this camera on, right?
We are not able to see you.
We can see you. He's there.
Oh, okay, sir. I think it's not there on my screen.
Yeah.
Yes, sir, please.
Yeah, thank you, ma'am. So good evening, everyone. So today I come to the concluding day of the 5 days long AICTE sponsored FDP on artificial intelligence in drug design and discovery, current and future perspective. So for today's valedictory function, we have with us our beloved, campus director, Dr. Manish Paul S., who is alumni of Harvard School of Medicine and discovery scientist. He has few dozens of patents to his credit, and he's also co-inventor of India-made antibiotic that is enmetazobactam. So I welcome you, sir, for the valedictory function. Dear faculty members, you all listen to various talk by a galaxy of scientists and experts on artificial intelligence in drug design, healthcare, and related area. I hope you have all enjoyed and have learned a lot. So objective of this FDP was to sensitize the faculty members to the artificial intelligence in drug design and pharmaceutical field. I hope this FDP was successful. So I request you all to encourage your students to take up some training program on machine learning or deep learning and data mining, which is the part of this artificial intelligence. During this B Pharma M.Pharm course, which would really surely add value to the program and to the career. So on behalf of management and faculty members of Acharya and B.M. Reddy College of Pharmacy, I thank all the distinguished speakers for sharing the expertise with the delegates and making this event so grand. I thank all delegates for participating in this FDP and make the event a grand success. I thank on behalf of Acharya Institute, AICTE, for supporting this FDP with the financial grants and the management of Acharya Institute for all the support and encouragement. I'm really extremely thankful to the organizing team led by Dr. Sajeev, Dr. Madhavi, Dr. Venkatesh, and other faculty members of Pharmaceutical Department of our college for organizing the event so effectively and flawlessly. I request all the participants to give their feedback about this FDP, which will really help us to improve. Thank you. Thank you once again.
Thank you, Amit sir, for your kind words. I now request our chief guest, Dr. Manish Paul, who has already been introduced by our principal, sir. And I would like to add that Dr. Manish Paul is a scientific advisor to various pharma and biotech companies and has to a considerable extent contributed to the field of science and is now leading us with his expertise and experience as our campus director. Sir, over to you for your kind words.
Thank you, Dr. Madhavi, Dr. Sajeev, and Dr. Amit. Well, a small correction I'd like to place on record. I'm an alumnus of Johns Hopkins School of Medicine. So that's one correction in my introduction. All right. Thank you. Well, it's been a great privilege that you all have had, and I too have had the privilege, especially the first day, to witness the inaugural session of all the speakers. It was a brilliant session. I think people spoke their mind, and I didn't attend the rest of the program, of course, other than the inauguration. It's a well done program. I have seen the speakers and the content. And of course, this is a field which is very interesting because I always believe in exponential technologies and how we adapt them. Because the traditional path of drug discovery, especially and development, coming from that domain, it's very dear to me. The traditional path, of course, is very lengthy, expensive, and does suffer from high failure rates because we have to test millions and millions of molecules. You think of a drug discovery, it's like a funnel, is what we are used to. Thousands of millions we screen, and only few of them come out as successful molecules. But however, since only few of them come to preclinical stage, and then they go on to hardly one or two come to the clinical stage. Having put a molecule from ideation to the clinics, the journey has been 15 years long. And unless someone has an appetite and that kind of a money, no company would fund a discovery program. So embracing innovation, particularly the automated technologies, is very essential to reduce this complexity associated with drug discovery. And it also circumvents the high cost, right? And the time we spend in bringing a drug to market. Then further, we also need to incorporate automation, which can make the hunt of the drugs quite cheaper. It's at least $1 billion which goes in before a drug can come to the market, and we do not know by the time the drug has any relevance at all, right? The risk is very high, but the rewards are also equally high. So we have to incorporate these modern technologies. And the past few decades have witnessed a dramatic growth in the use of novel approaches and technologies in drug discovery, especially the bioinformatics, the molecular modeling, and these things have really taken it to the next heights. Further, the availability of massive data sets, right? Today, data is the new gold. This and the advanced algorithms has also driven more interest and major improvements in the use of AI, especially in the field of drug discovery. But of course, AI does provide substantial improvements at many stages of drug development, reducing the time from target identification to clinical development. And there is another subset of AI, that is the machine learning, right? That is also rapidly evolving. And you heard speakers speak, and I know Ajit was there, our colleague from Acharya. And this field is increasingly being harnessed by many pharma companies already. So integrating ML approaches into the drug development process does help to automate a lot of repetitive redundancy of data processing and analytical tasks. So I personally, being a champion of AMR stewardship, right, antimicrobial resistance stewardship, especially in this week, which is, we call it as WOW, not VWOW, it is WAAW, the World Antibiotic Awareness Week. Right? In this week, we talk more of the role of antibiotics and how we preserve and protect these antibiotics. Right? I'd like to stress that the rapid emergence of antibiotic-resistant bacteria is a matter of global concern. If there is any urgent need, it is the discovery of antibiotics, because it is predicted that if no action is taken, then the resistant bugs, right, could be responsible for at least 10 million deaths each year by 2050. And we also read, I think a couple of weeks ago, the data in the newspaper stating that the fertility rate in India is also falling down. I read this, the headline says, the fertility rate in India is stabilized, right? That's very interesting caption to read, which means things are already reducing the number of people on Earth holistically all over the world. In addition to that, if we get infections, I think we become visitors on Earth rather than actual people who live on Earth. So further to this, I'd like to add one more point to everyone's attention who has participated in this program, and that is quite dear to me of late. I've been talking a little bit more about this particular topic, that is superintelligence. We always talked about artificial intelligence all this time, but I think it's time that we start focusing on superintelligence, because superintelligence is a word coined by Nick Bostrom from Sweden. And what it means is how our neural network of our human brains, and when it is supported with the AI driven, the machine technologies, how we can make an exponential leap in the way we look at the challenges and we address those problems. So I would encourage all the participants around the country are there here, as I was told. I would encourage people to think in these lines of how can we harness the new technology, practical applicability. One is, of course, many of us do come for programs to get our, an experience of networking, to learn newer topics, and then that's where the shop closes. But I would encourage everyone to start applying them in their practical life. Write programs, programs in the sense, I meant the project proposals which incorporate the modern technologies. This brings in a transdisciplinary approach. We bring in people from commerce, the economics, because economics drive the country, the world, right? Economics, the technologists, the computer scientists, right? And also the pharmacologists and biologists come together. We can make a significant difference. So I would encourage everyone to use this platform as a continuing, a stepping block or a bouncing board to take this entire program to the next orbit. Wishing you all the very best. Thank you very much. I sign off.
Thank you, sir.
Thank you, sir.
You're welcome.
Thank you for joining us.
Thank you, Manish sir. We have effectively summarized the whole AI, the concept, and instigated and what has to be taken next. So data is the new goal, and we had one of the speakers says that right now we need new possibilitarians. Yes,
I like
and
the
I
word.
believe.
Can you please repeat
Yes,
what's the word?
Possibilitarians.
Wow, that's a good one.
And that I think now, as you were coining it, that we need possibilitarians for new antibiotics now. Let's look forward to that. Thank you for your words, sir. I now request our FDP convener, Dr. Sajeev Kumar, to present a brief summary of the FDP and deliver the vote of thanks. Over to you, Sajeev, sir.
Thank you, Dr. Madhavi, for inviting me for delivering the vote of thanks. First of all, I should appreciate the whole and entire team of Acharya Institutes, who has hand-to-hand worked together to bring up this event in a very smooth manner without any flaws. That's highly appreciated and has been given as a feedback by the delegates itself through WhatsApp and through by all means. So I congratulate the entire team who are behind this whole show to take up for the vote of thanks. Very good evening, one and all. Today we have come to the closing session of AICTE Atal FDP on AI, Drug Design and Development, Current and Future Perspectives, organized by Department of Pharmaceutics, ABMRCP. My applause to AICTE Atal New Delhi in recognizing our institute to initiate faculty development programs and in providing the necessary financial support. As a program convener, it's my privilege to propose the vote of thanks on the day of this event. I, on behalf of Acharya BM Reddy College of Pharmacy, Bengaluru, would like to thank our distinguished guest, Dr. B. Suresh, Pro-Chancellor, JSS Academy of Higher Education and Research, Mysuru, for inaugurating the event. I would like to thank the guest of honor, Dr. Shankar K. Pal, for accepting our invitation and in joining us for the event. My sincere appreciation to all the speakers who have accepted our invitation and sharing their valuable thoughts and making the event possible in time within a short period. Our kind recognition to the management of Acharya Institutes for their constant and continuous support in organizing the FDP. I would like to thank Dr. Manish Paul S., our campus director, Acharya Institutes, and the chief guest for this particular valedictory function. Thank you, sir, for accepting our kind invitation and joining with us on this occasion. His leadership capability, zeal, and instructive thoughts has led our institute to march ahead and to excel in all bands with much recognition and glory. My sincere thanks to our beloved principal, Dr. Amit Kumar Das, for his constant support and encouragement and in guiding us in forefront. I take this opportunity to thank all the delegates who have participated in this FDP actively and unveiled their interest to enrich their skills and knowledge in the area of AI. My kind recognition to our Vice Principal, Dr. Manjunath PM, my teammates from Department of Pharmaceutics, Dr. Venkatesh DP, Dr. Madhavi BLR, Dr. Joyce Ruby, Mrs. Ashley, Mrs. Chaitra D, and other head of the departments and other faculty at ABMRCP. for their untiring support and coordination to organize this event smoothly. Once again, I thank each and everyone for being part of this workshop and making it a grand success. With these warm words, we move to the end of the 5 days FDP. Thank you all for joining with us and wishing you a great day.
Thank you, Sajeev sir, for proposing the vote of thanks. Before we close, I have a few instructions for the delegates. You can all turn on your cameras so that we'll have a screenshot. The cameras are permitted. We'll be glad if we can, at least a few of you, if we can be accommodated on the screen. Thank you so much. It's been wonderful to have you all here. I request the technical team to do the needful with regard to capturing any necessary screenshots. And a few instructions for the delegates. Post FDP for any queries. Please send an email to fdpacharya2021@gmail.com. CC to principal abmrcp@acharya.ac.in. CC to your respective institution principals. No other kind of communication will be entertained. This is just for your information. And may I know if the screenshot has been captured? And the responses have been closed for the assessment. This is for your information. I'm sure the FDP was quite enlightening and first letting us some awareness where we are lagging behind. Those who are able to switch on your cameras will be glad to have you on screen. So, can anyone, technical team, can you please confirm if the screenshot has been taken?
Done.
Yes, it is done. Thank you all for being with us. And before I formally conclude, we are all much beholden to the convener of this FTP, Dr. Sajeev. For him, this has been a Herculean task, trying to pull up speakers from all across. And I should say the gestation period has been more than 9 months, having taken this up, I think, in Jan 2021. Sir, thank you so much for convening this FDP, for the proposal, for the acceptance. And now we are glad that we have successfully completed this 5-day ATAL FDP on artificial intelligence. Thank you one and all for being with us.
Well done, well done.
Thank you, Amit sir. So now I conclude this 5-day FDP and let us all raise for the